Okay, so welcome to UCLA QC Bio Workshop 36, Advanced Unix for Scripting and Queuing for Large Data for the spring quarter of 2023. This is a new workshop. It's still kind of not quite sure exactly how this is going to go and so on, but we'll give it a try. I need to flip back and forth a lot between the slides and a terminal window. So here's going to be a Hoffman window. So that's why I'm not going to do the slides with the full screen. You should have an email from this morning, I think, that should have a copy of the slides. And if you want to try to follow along, it's not completely trivial to follow along because there will be the data files, which are in my scratch space in Hoffman. And if you want to really literally follow the command, you're going to have to copy that stuff to your place. Otherwise, you're going to have to change paths and stuff in places because you can't write into my script. But some of the things we'll do will also not depend upon the data. I mean, to do this, we need to have some large data examples. So there's almost 100 gigabytes in the data files that are in the examples. If you do want to copy them, just be aware that they're big. They're not a full blown, like when I say large data here for this workshop, I mean, this could be, you may have a terabyte project or something like that. I don't want to have something that big as an example to do live because it'll be too slow. But 100 gigabytes is still pretty decent size. It's not like little trivial examples. It's actually something that kind of will illustrate all of the things that would go on even with a terabyte scale project. Okay, so yeah, so it's really amazing how computing has sort of like scaled up over the last 50 years. I just turned 50. And over my lifetime, CPU has probably gone up by about a factor of 10 million, memory by 10 million, communications, networking, 100 million, and storage like disk, where you put your files, like a billion-fold. I mean, it's really kind of amazing. And that is really something that biology takes advantage of right now because ultra-high sequencing, which we can invent so many applications, we can adapt so many things to basically turn a biological question into sequence a whole pile of DNA and then analyze that pile of DNA to answer the question. We have so many different, we even have like these cool like spatial, single cell RNA sequence, all this kind of stuff that was just sort of science fiction until fairly recently. And compute is really what makes that happen. But the problem is you have to tell a computer what to do, right? So now there's like ultra mature applications which are rarely seen in bioinformatics, but this is stuff like Adobe, and Microsoft and so on. These are things that are, canned software, broadly applicable, very, very mature, very polished, doesn't need much technical demands from users. We're almost never in that situation in bioinformatics. Sort of the next level is sort of like either mature applications that have been around so long they've kind of like completely stabilized, or compromised applications like Galaxy, where it's sort of like not quite as flexible as really doing everything on the command line and so on, but it's a nice GUI interface that lets less sophisticated users get work done. And some bioinformatics applications are in there, but most of the time of the day-to-day bioinformatician is not at this level either. This is maybe more wet bench people spend their time here. Then the next level is maybe kind of things where kind of you're not going to write code to do like the actual alignments with like with the like suffix arrays and so on or so on. The heavy lifting is going to be in programs that people already wrote, but you need to glue them together because you're going to have an analysis that uses this program to do this step and this program to do this step and so on. But you got to glue them all together. You have to compare inputs, take outputs from one, transform them into inputs for the next, and so on. And so you need some glue, and oftentimes the glue is going to be shell scripting. And so Bash is the default shell on Linux, and that's the one I'm going to talk about. And then you have the whole plethora of standard Unix POSIX utilities, like awk and set and cat and grep and all those and so on. And those are all very simple utilities individually, but even with the glue that shell lets you do, you can put them together and actually achieve fairly sophisticated things. And that's the level we're going to really focus on in the workshop. Here's a list of some of these you'll recognize, some of these you won't. But these are sort of like the common commands, utilities, and these are all the shell commands actually. Then you reach levels where sort of like you actually have to start doing some programming. and then into your Python, Perl, things like this, various other scripting languages. And then that's if you kind of prefer sort of lighter environments. If you want heavier environments, then maybe you do R or MATLAB or Mathematic or something where you have you're really in sort of like a graphical environment with huge libraries behind you and so on, lots of stuff built in and so on. And then only the most demanding applications where you sort of really are kind of like at the edge of what your hardware can do and so on, either in terms of memory or speed or something, then you're into the low-level systems languages like C and C++. And that's actually the level that underpins everything, because what's Python written in? Well, Python's written in C and C++, and so on. What's Firefox written in C++, right? It's all at some level that's the bottom. But most bioinformaticians probably don't spend much time down there because we prefer, we prefer most of the time you're doing things that are just adapted to the project you have at hand, and you can use other tools like Bowtie and so on that are written at that level to do kind of the core operations that do need that level of performance, and you can focus more on gluing things together and so on. So these are the levels, and generally to achieve a project, you generally try to use the kind of the topmost level in this, the bluest level in this order. That gets you the job you want done to the quality that you want it done, and uses reasonable resources to do it, right? So as long computational resources are generally cheaper than human resources, so you generally kind of go up to the blue side as much as you can. That's your trade-off. So for most bioinformaticians, we're in here. We're in this level, somewhere between shell script and R, say. Here in this workshop, we're going to focus on the shell script level. Okay. All right, so why learn more shell? Okay, and in the slides, there's actually a bunch of kind of links. Some of them are kind of Easter eggs, they're hidden around, but if you click some of these, you will go to some interesting web pages. Like here is a very nice list of shell stuff, and in particular, some of these guide links at the bottom have very nice stuff about shell scripting and so on. You can look at those when you have time. So the shell is nice because it, first of all, it's the canonical top-level interface to command line environments, and it's always there. So, and even if you are using these other programs written in Python or whatever are to do your work, the actual work, you still need to get files into them and manage files and transform inputs, to different formats and stuff. And that's always kind of shell is always kind of the place where that happens. The environment of shell and the standard utilities are very reliable, very portable, and very powerful. And so they let you get real work done pretty much anywhere. On a Mac, you're using basically a BSD UNIX system. You've got a built-in terminal. In fact, the default terminal until recently was Bash. It's ZSH now, but ZSH is mostly Bash compatible. If you're on a Windows machine, you can install Cygwin or the Windows subsystem for Linux and have a full Linux-like environment there and use the same. You get a Bash shell and all the utilities. And if you're on a Linux machine, whether it's a small machine of your own or maybe a machine in the lab that's a little bigger, or the Hoffman cluster on campus, or a gigantic supercomputer like some of the ones run by the national labs and so on, which also lets you get time on them if you need. Same set of commands and everything there. So everything you learn is transferable to sort of all these environments you're going to be likely to ever use. It's been around 50 years since the beginning of Unix in like 69. And it's still largely compatible way back to the beginning. If you don't use some of the features introduced later, yeah, I mean, if you use a new feature, you got to use a shell that knows the feature. But basically, it hasn't. I mean, it's sort of maintained background compatibility for much of that time, and it's not going away anytime soon. And a lot of people spend a lot of time in the shell. I kind of looked through my own. I've done bioinformatics for 20 years now, and I've written. I went and looked, and it's like I've got tens of thousands of shell scripts, and and so on. I've probably written more shell code than C code and Perl code and so on combined, because it's like it's sort of the glue that's part of every project, and it just it always adds up. It's very conducive to gradual learning. So you can get started. That's like our W1 workshop, Intro to Unix. This is the sort of the next course after that. And even with just a very small set of stuff, you can start to get stuff done. But then, I mean, it's very hard to just sort of like learn all the shell all at once. That doesn't really happen. The way you learn shell is you reach situations in your actual work that would be nice to have some feature. Then you go and try to figure out how to do that. And you learn, oh, this feature exists, and then you implement it in your current application, and you learn kind of as the need comes up. And so the way we're going to learn about things in this workshop is we're going to have basically a case study. So we're going to study kind of one bioinformatic problem and then kind of work through it. And as we work through it, we'll reach situations where sort of various features of the shell will come up naturally and be kind of cool and will help us efficiently work with it. So that's kind of the way it goes. You kind of learn it gradually. And it's actually pretty efficient. So efficiency can mean two things, human time efficiency. How long does it take you to express to the machine what you want done? And computing efficiency, is the memory and RAM and disk and network usage reasonable? Actually, the shell can often be pretty decent for both of those. It's very easy to throw things together with like pipelines where you start taking outputs of one command, feeding it to the input of another command, and so on. And when you get good at pipelines, you can have actually all the parts of the pipeline working at the same time. It's not like you have a gzip file, you unzip it to disk, and then you do something with it and get an uncompressed file, and then you gzip that output file, and then whatever, and you do all these stages intermediately. You learn that actually you can just get rid of all the intermediate stages, run them all at once, and the data will flow around to all the programs working in parallel, and it's much more efficient. And then the way shell works is it's fairly easy to express that. And so that ends up being quite computationally efficient because it's very easy to run things in parallel in the shell. It's sort of the natural thing that happens when you do pipelines in the shell. And you can also get rid of a lot of redundant I/O because if you do things in parallel pipelines all at once, the intermediate stages are just flowing through RAM and stuff, and they're not going back out the network to permanent storage and back again. Also, a lot of the stuff you do in the shell is in its streaming mode of computation. So with very big files, oftentimes the way you work with them is that you cannot read them into Python or R or something all at once because they don't fit in memory, right? So if I have a NovaSeq S4 lane, might be 500 million reads or something, that file is going to be many gigabytes, and uncompressed, it's many, many gigabytes. You try to load that into RAM on Python or something, you're going to run out, and that's a fairly big machine. And then you're going to want to be able to do things with it. You're going to have a couple copies in memory and so on. Very quickly, you end up with, unless you're kind of a rich guy, you don't probably have a 0.5 terabyte or a terabyte RAM machine, although those exist. So in a shell, oftentimes the way you work with things is in a streaming mode. You open something, the data just comes out in one line at a time or something. You work on it as it comes in, and as you process it as it comes in, and then you're also running the output as it comes in. And at any one moment, you're not really holding very much in memory at once. Just sort of, things are streaming in, you're processing them as they come, and then they're streaming out. And that mode of computation, which pipelines are very adapted to, allows you to work with files of essentially infinite size because they don't have to all be in memory all at once. And so that's very powerful with a lot of bioinformatics file formats because a lot of them are plain text, either directly or convertible, like BAM files for alignments these days are really SAM files. SAM files just binary compressed. SAM files are plain text line-oriented format. Very easy to turn a BAM file in a stream dynamically, leaving the BAM file on disk into a stream of text that you can use in a pipeline using SAM tools, say. And so you can work with 500 million read files, no problem in the shell. Whereas if you're not going to open an entire BAM file in R, say, and read it all at once, and then try to filter it and do things with it or something. So this workshop, I'm going to assume you've already taken W1 or have experience equivalent to W1. And so you have a Hoffman account because that's where we're going to do our examples, and that's a great Linux environment on campus, and it's also an environment where you have a lot of compute, which if you're interested in large data, you need a lot of compute. So Hoffman 2 is going to be where we're going to do our examples. And you've seen basic uses from workshop 1 level stuff using these kinds of things, ls, cd, pwd, this kind of thing. And you know a little bit about pipelines. You've seen some simple input and output and pipeline redirections, and so on. So originally, when I kind of was putting this workshop together, I was like going to try to like cover all of the things, like all the shell features. And by the time I got to like slide 60 and I was at like 2% of the shell, I decided that was impossible. That wasn't a good way to do it. So one of the things is about being a 50-year-old system is that it's a system that people live in their daily lives, is that it's accumulated a lot of features because everybody kind of wanted to add something that was going to help themselves out. So the whole thing is kind of organically evolved and very big. Practically, when I showed you this list of punctuation characters, almost there's like a ton of shell magic that's hidden in combinations of these punctuation characters, and like practically all combinations of these things mean something. But you don't really need, like I said, it's something that you don't really need to know all of to kind of start going. So that approach wasn't working too well. So the new thing is to do sort of a case study. We're going to study a particular problem, and in that particular problem, we're going to have some nice things arise, and we'll talk about them. So this particular example will be a large alignment problem. So the file size will start big. But the way you're going to handle a large alignment problem in Hoffman, the most efficient way to do it is to actually cut it into lots of small pieces. Because we'll talk about that, because that's the way resources work on Hoffman, is that single large jobs don't work very well, but zillions of small jobs work very well. So we take a large alignment problem, break it into a zillion little alignment problems, and that'll turn a large file size problem into a large file count problem. And then we'll talk about how you might process such a thing, either working on a single computer, Mac, Linux, or Windows, because we talked at the beginning how you could have the same, the kind of the standard bash and utilities environment at any of those platforms, or equivalent to that, a single node or interactive session in Hoffman 2, that's equivalent to basically working on a single computer. The commands would look the same. Which is really what you're going to want to do once you get into the large data world, how you would do that with the Hoffman 2 job queue system. Because even if you've seen kind of basic uses of the job queue system, those don't scale too well to, say, thousands of jobs, which is what you're going to need to do when you have a big problem. And so we're going to build up to the way you would successfully handle large numbers of jobs, large meaning now thousands of jobs on Hoffman. So I don't know if we're going to fill all 3 hours of all 3 days. We'll see. So I'm not going to particularly rush because it's not really important exactly where we end up, but more that you get something out of this. So, ask questions if things aren't making sense to you and so on. Ask questions and so on, because I'm trying to help you guys, and it doesn't serve anybody any good if I just up here and talk and nobody understands anything. Whatever speed we need to go at, we go at, and wherever we end up at, we end up at. And the slides probably won't have anything, because probably to explain things, we're going to have to do some interactive, experiments and so on. Like when we talk about shell arithmetic, we can start doing little experiments like this. Those won't be in the slide, but it's easy to explain things like this in an interactive way in the terminal. Okay, so if you haven't already, log into Hoffman. So, okay. So I have passwordless SSH enabled. That's very convenient to set up if you haven't set that up before. We can talk about that after if you don't know how to do that. And then, you're not supposed to do anything really on a login node besides use that to switch to some other nodes. So let's get on a compute node. And that already brings up the question of how much resources to ask for our interactive session. Now, the usual problem on Hoffman is that the more resources you ask for, it's a smaller fraction of the cluster that could ever satisfy the request, a fraction of that can currently satisfy the request due to the currently running jobs scattered across the thing. And then there are things like your priority and whether the group you belong to has bought nodes on Hoffman and so on, kind of fudge factors. But basically, yeah, as you increase your resource requests, you reach a point where the waiting time to get those resources goes up and up, and eventually it goes to infinity in the sense that you're never going to get it because either the cluster doesn't satisfy that or just the whatever. So So here I'm going to ask for a rather small amount of resources, but hopefully one that like even you guys will have no problem getting interactively in a few minutes. So we're going to ask for we're going to work on a single node. So we're going to ask for 3 cores on one node, 4 gig of RAM per core, so 12 gig RAM total, and we're going to ask for 3.5 hours so that it covers our 3 hours today. So I'm going to put those in some shell variables, right? So hopefully you saw in W1 shell variables, right? Now those can get very complicated, but in their basics, pretty simple, right? You can assign a variable, whatever, you can use a variable, whatever, and so on. And shell coding is probably another thing I should say a few words already. The shell is a very string oriented system, text processing kind of system. And that means you're always got problems about trying to communicate exactly how you want strings broken up into smaller strings efficiently, correctly. So one of the biggest problems, say, is white space. So is white space separating different strings? We know that commands take arguments, right? So, so here's my files, or whatever. So, commands take arguments. So, here's just some file. I don't know what these are, but so there. So that's actually running a command called ls, which is actually a binary executable file on the file system called user bin ls. It's taking 3 arguments, one of which is -la, one of which is this, and one of which is this. So ls is getting 3 arguments. ls happens to interpret this argument special because it starts with a hyphen, and then ls supports all these options that change how it runs, which are all documented in its man page and so on. But suppose I had a file name that has a space. I'm going to switch to my Scratch directory. That's a little cleaner than this home directory. So let's use Scratch C Focus, and then the files for today are inside this directory, Advanced UNIX Workshop 2023 Max. Okay, so this is the place you should be to see the files or to copy these out to work on your own. Your own Scratch space will be whatever it is. So if you don't know where your Scratch space is, it's here. That's mine. So yeah, so here's some files that are relevant to this workshop. So suppose you can actually make files that have spaces in the file name and so on, right? Well, so now you got a problem, right? So now suppose I try to at that file. Well, that's not right, because now you've got this problem of trying to communicate, is A space B 2 arguments or 1 argument with a space in it? And that's where quoting starts to come into it. So I could use single quotes. This is an empty file, so let's there we go. Now we have something, a non-empty file. So single quotes are very powerful quotes. They turn almost everything off except single quotes. I think they turn off literally basically everything except single quotes. \new line might be an exception line continuation. Less powerful are double quotes, which will certainly protect white space. So that'll be okay too. Both of these times, cat is getting a single argument, which is 3 characters long, which is A space B. But double quotes allow a lot of shell stuff to still happen inside. So you can still get shell variables to expand inside single quotes. So if I have some shell variable here, that still expands in a double quote, but it won't expand in a single quote. That's probably about all we need to know for now. Double quotes will also quote single quotes, no problem. And of course, single quote will quote double quotes, no problem. How would I get a single quote in a single quote string? I would have to terminate the single quote string, get a single quote in there, which you could do like that is one way to do it. Or / also turns off the special meaning of characters. So that would be a single quote. And then I could go back to single quotes if I wanted. That was embedding a single quote into a single quote string. Anyway, so that's probably all I want to say right now for quoting. But anyway, I'm going to use double quotes and single quotes a lot, even where not strictly necessary, but it's kind of a good practice to get into that things that are logically one unit, like file names, which could have spaces in them and stuff, and that you want to keep as one unit, you should generally have double quotes around most things, unless you sort of know that you don't want to preserve white space. So anyway, so that's why I sort of maybe a number I won't double quote because that's really obvious that it's not needed. But strings I sort of automatically double quote because just just have to don't even think about it then. Okay, anyway, so here's the little qrsh command to get us an interactive session. I just put echo on the front so we can see what this expands to. So you can just see it substituted in the current interpretation of how many cores I want, and here's the runtime, and then here's this, and then here's a little magic, because on Hoffman, if you read the documentation, when you ask for PE shared to get multiple cores on one node, you're supposed to do your resource requirements with H data being the memory per core, but you're also supposed to say HVMEM, which is the total amount of memory for obscure reasons. But this is the way the Hoffman documentation actually said you're supposed to request resources on Hoffman. Now it's nice to, if we didn't actually have to multiply it out, right? So I don't have to do a hard-coded constant 12. I can just have the shell do that computation, right? So one of the little bits of shell magic is $ with double parens does arithmetic, integer arithmetic. On most platforms today, 64-bit signed 2's complement integer arithmetic. So that lets you do things like 2 times 3 and whatever, or here we're doing 3 times 4. But if you just use a shell variable name, it just looks those up anyway. So there we go, same thing. Anyway, so that allows us to sort of, whenever you can, you kind of try to make your scripts so that they sort of require the least changes as possible to make flexible. So here is just a very simple case where sort of like you just use shell arithmetic to stop you from having to maintain the relationship of 2 numbers being multiplied to another number. Anyway, so let's actually run this now and get our session. Yes, so you start on a login node, and login nodes, you could compute on them, but the point is that if people started computing on a login node, since that's the bottleneck that every person that uses Hoffman has to go through, if you make a login node slow, you're hurting everybody. So the idea is the only purpose of a login node is to immediately use that to go to another node, because when we go to, here I got node 1058, you guys will get some other node most likely. And so now I'm using a machine that the scheduler has scheduler has blocked out 3.5 hours on this machine. 3 cores, 3.5 hours. This guy with 12 gig of RAM, that's his now. And the scheduler now has this hole, and the scheduler will now schedule around this hole. And so now we have, we're being nice. So the login nodes, essentially their only purpose is to be sort of a network intermediary from the outside to the inside thing. They're just passing network traffic in all the time, but you shouldn't be running any commands really on a login node. So the nodes on Hoffman are very heterogeneous. It's not like they bought 10,000 copies of some HP server that are all identical. The nodes on Hoffman are on a rolling schedule, and so as we buy new nodes, the campus buys new nodes. Those are bought with more recent hardware. And so the sort of the RAM and the number of cores and stuff like that kind of grows over time. But the lifetime of a node is 5 years or something. So there's sort of a whole scattering of nodes across the thing. So in general, they're usually pretty beefy. I mean, you can start using some of the Linux stuff to figure it out. So like I'm on node 1058. How many cores does this thing have? Linux, you have to know a little about Linux, but if you look at proc CPU info, you'll see that this thing actually has the first processor 0. So this actually is a 48 core machine. How much RAM does this thing have? Proc mem info. Well, it's in here, mem total, blah, 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 kilobytes. Oh gosh, so there's a comma, there's roughly a K, it should be 1024, not 1000. But anyway, that's KMG, 256 gig of RAM. So this is a 48 core, 256 gig RAM machine, and this is some random machine I got. But that's probably near the upper end of memory. This is probably one of the beefier machines on Hoffman. But so you could make a resource request for give me 48 cores and whatever 256 divided by 48 is. What is it, 5? You call it 5. You could do 48 cores, 5 gig of RAM, and you might get this machine. But you are now asking for this entire machine. So now the scheduler has to now start to work that. Anybody else sharing this, anybody else with processes running on this node now, it has to sort of clear out, right? So, and get it empty. And once it's empty, then it can give you the machine. But you may be waiting a day, or something, right? Because I think the default maximum runtime you can ask for is a day. That you could wait a day because the scheduler's got to work at emptying something out to get you onto a whole machine. So yeah, so anyway, so this gets back to the point I was trying to make earlier, which is that, yeah, this is why it's better to not try to run jobs as small numbers of huge jobs, but to cut them up into tiny pieces, because the scheduler can put tiny pieces in places a whole lot easier, right? So if I've got, like 1 gig RAM jobs that need 1 core and are going to run for an hour, the scheduler can just put those all over the map, because it basically can fill all the holes it has right now with those things. And so you can typically get hundreds of those running at an instant, like in a moment, with no problem. Whereas if you start asking for, oh, give me 100 gig of RAM and 30 cores, well, there may not be many machines that ever can satisfy that, and they're probably all partially busy right now. So now you're waiting a long time. So this is actually the motivation for why we want to split the data up into small pieces. And then that motivates dealing with huge numbers of jobs. And then that motivates this whole workshop. So yeah, okay. So all right. So now each, we just SSH into it. We basically just SSH into node 1058. It's called QRSH, but it's basically SSH. So that's actually a new shell. So that gives us our default, directory and so on. You notice none of the environment will carry over. Each time you make a new shell or run a shell script or something, they're all getting a fresh environment. So that's why I'm back in my home. Oh, I think I already too fast. I think when we started the session, it was in the home directory. I already switched back my scratch space. Anyway, so if you haven't yet, you got to switch back to your scratch space or my scratch space, wherever you want it to be, and move back into the workshop directory. And maybe I'll get rid of this AB file. We don't need that one anymore. Oh, another exciting thing about shell quoting is, so don't make files called like this. It's even, there's even a worse example. So now I have a file whose name is -rf. Now, if I start doing like rm star, what do you think is going to happen? Or rm, rm star f or something, right? That's going to include this probably as the first file, because hyphen sorts very early in the alphabet. And that rm is actually going to interpret as an option. And r means recursive and f means force. And rm will now delete everything. This would all go away, even if you think star f should only match a few files here. So one of the things quoting you also got to worry about is things, you may think of something as a file name, but that doesn't mean that the command's going to think of it as a file name. So that leads you to all kinds of things like double dash. So a lot of programs accept double dash as an option, which means after this double dash, there are no more options. So that allows you to do things like have file names with leading hyphens, because you've now told RM everything past this point is definitely not an option. But anyway, the real thing is you really don't want to create files with leading hyphens, because it's way too easy to have programs interpret those as options. And those are just more hassle than they're worth. If you're writing scripts that have to be robust and you don't know what's going to get passed in, then yeah, there's all kinds of security problems and so on with that. But most of the problems a day-to-day guy worries about are things like white space in the file names, and not so much leading hyphens in the file names. But just to kind of give you an idea of how bad sort of the quoting problems can get and so on. It's more than just quoting. It's an interpretation problem in general. So now I got to get rid of this file. So actually, though, that's how you would get rid of this file. There we go. So now it's gone. You saw my first attempt to actually create that file didn't work. Touch creates an empty file with whatever name you give it, like there, no problem. My first attempt was this, which doesn't work because touch itself takes options, and it was trying to interpret the RF as an option. So that's why I had to say this, because touch also understands the double dash. Anyway, that's too much time on that. But okay, we're only on. We haven't gotten anywhere yet. We basically did one command CD and we will have a long discussion, but that's okay. Okay, so yeah, so here's going to be the case study we're going to do. So we're going to do a whole genome bisulfite example. Here is the paper. You can look at this when you want or whatever. It was a Nature paper quite a few years ago now, whatever. But it's basically studying DNA methylation in a bunch of human stem cells induced to various things. So the two samples we're going to look at are H1 ESCs differentiated to Bezendoderm, 2 bioreps, 1 and 2. These are in NCBI SRA. There are actually 5 runs for biorep 1. So those are the SRR sessions. There's 4 runs for the biorep 2. That's these 4 SRR sessions. I merged those. Those are what are now under the NCBI SRX identifiers. So this SRX is BioRec1, and this SRX08 is a BioRec2. These are 450 million reads and 350 million reads, 101 nucleotides, single end, whole genome bisulfite to human, obviously. And we want to align these. And more specifically, we want to disulfite align these. Now we don't care about the biology here, right? So we're not going to really care. We're going to use BS-Bolt to do the BS alignment and stuff, but we aren't doing this because of the data or the output of this alignment or whatever. We're just studying this now as a computational problem. Okay, but one of the primary parts of a whole genome bisulfite analysis would be the BS alignment stage, right? And because whole genome bisulfite requires deep coverage and human is a big genome, it means individual samples tend to need huge amounts of data. So that's why we're blowing like a whole S4 lanes, today's equivalent, on one sample here, 415 million reads on one sample. Compared to say like an RNA-seq experiment where maybe you might have 20 million reads for a sample. So WGBS individual samples tend to have huge numbers of reads anyway. Then you may have 20 samples or whatever. Right here, I'm just going to do 2 samples, and that's enough to be an example. But you may have 20 samples. Furthermore, bisulfite alignment tends to be quite a bit slower than ordinary alignment due to the ambiguity. It's essentially you've gone from sort of 4 bits of entropy per base, 2 bits of entropy per base, which got ACGT, but now the C to T transition is confounded with the bisulfite conversion. You're down to sort of like a 3-base alphabet, and sort of you get less entropy per base, and things. It's more, you have to work harder to kind of get the alignments done. So the alignments themselves tend, on a read per read basis, tend to be slower. So combine those 2 factors, and now you're talking about this is way slower than like aligning a bunch of RNA-seq stuff. So a good way to handle this task then is we're going to take the FASTQ read files for each sample and break them into a zillion pieces, BS align each piece as a large Hoffman job, but will really be made out of zillions of little subjobs that the scheduler will find very easy to place. And then after that's done, we have to combine the files back together again so that we get single BAMs per sample, like as if we hadn't split them up. Right? So that's basically the kind of the computational flow we're using this example in the workshop to illustrate the problems that occur with large data. So we'd like to just be able to essentially run BS-Bolt on the whole file with one command very easy and so on, but that's going to take too long. We couldn't even do it because we can't even get a resource request that big, and so on. So that's what we want, but the way we're going to do it is cut the pieces, do the pieces, merge the pieces back together. So we're going to end up at the same point, but in a way that's both computationally feasible and actually the fastest way you're going to be able to go to Hoffman anyway, even if resource requests weren't a problem, because we're going to be able to throw so many cores at the problem, because the alignment speed is going to essentially be how many cores do you have working on the problem at one time. It's going to control the wall clock time, right? Even if you had a big machine that could grind through it all in one pass as one part instead of in many parts, that machine may be 48 cores or something, right? And that's as big as machines get. But if we cut into little pieces that we can use multiple nodes, we'll get situations where you could have 500 nodes working on your problem at one time, maybe 1,000 cores, 2,000 cores working on your problem at one time. Well, that's obviously going to be a chance to make wall clock way shorter, because you just have the compute per second you're grinding through is just way higher in aggregate. So not only is it sort of like the only reasonable way to do it, it's much faster than the alternative would have been, even if you could do it. I mean, the only advantage to sort of the single approach is that you don't have to do any of the splitting and so on, whatever, and you just can run the one little command line that you're used to. You don't have to have all this infrastructure we're going to talk about and so on, and knowledge and so on. So yeah, you got to learn more to be able to do this, but it really helps you. It's more than just making the problem doable at all. It's about making the problem doable fast, and so it's actually the preferred way to do it. And this says the common situation in bioinformatics. Most bioinformatics problems are what are called embarrassingly parallel. And that means that the parallelization is trivial in the sense that the problem really is a ton of tiny, tiny little problems just over and over again. For alignment, every read can be aligned independently of all other reads, right? knowing where a read goes in a reference unit doesn't require knowing what the other reads are, unless you're doing something really fancy. You could have some learning process where you're actually learning that subsets of the genome are coming up or something, and then you use that to re-estimate sort of like ambiguous cases to where a read could go. I mean, you can invent cases where actually there's a feedback loop, and you kind of do need to know what the other reads are doing to know where a read goes. In line with it. Nobody, 99.99% of the line problems that you can find are not like that. The decision about where to place a read depends only on the read in the reference genome, not about any of the other reads anywhere else. And that means each read is essentially its own alignment problem. And that is an embarrassingly parallel problem, because if you've got 500 million reads to a line, you really have you cut that into 500 million problems and do that. Now you don't just cut it up that fine, because that would be too many. But you could cut them up into 10 million read pieces. Right now would be the opposite of embarrassing in parallel is completely serial. You can have problems that the data dependencies are such that the only way to do it is you have to do these steps in this order, and you cannot start the next step until the previous step finished. Then you can't parallelize at all. And there's nothing to do about it. Then there are problems in the middle where there's sort of some data dependencies, but it's complicated. And so then you have to really work hard to structure your code and so on to take advantage of the dependencies that are possible and so on. And that's where sort of like high performance computing comes in. And like the physicists are often in that situation. They're doing physical simulations of interacting things and so on. And so, particles are interacting with particles or something. So particles need to interact with nearby particles. And, so then, this kind of it's complicated for them. They have to work much harder to get their parallelizations done, because they need much more communication between the cores working on the problem, whereas we essentially need no communication between the cores working on the problem. Maybe the counterexample for bioinformatics, though, would be sort of like de novo assembly, or large RAM is really essential. You need a large machine. And maybe if you're doing like, there are some of these things now that use very large neural networks to predict protein structure. And those need large GPUs, large RAM GPUs, and there's no way around it. But most bioinformatic problems are not like that. It depends on what you're doing. But for most of us, most of our day is not like that. All right, let's look at the giant files we're starting with, okay? So here are the two primary input files. These are essentially as they come from NCBI. This is one sample, and how big is it? 39 gigabytes. And here's the other sample, 30 gigabytes. All right, let's just look at these a little bit to see what we got. We could look at one of them with less. That is the insides of a FASTQ file if you've never seen one. This is a read name here. This is the 101 base sequence. These have to be 101 base single line reads. And then for stupid reasons, NCBI SRA repeats the read name, which just serves to take up space, but gzip mostly compresses that back out. So not really a problem, but anyway, most FASTQ files just put a single plus sign here and omit the name a second time. And then this is the per base quality scores, usually Fred plus 33 ASCII shift encoding, which we don't really care about here, or whatever. But basically, it's a 4-line per read format. Okay, so those 4 lines are read 1, and then these 4 lines are read 2, and so on until you reach the zillion million lines that this file is uncompressed. Now, this is a gzip file. It's compressed, but less knows how to ungzip files when it shows them to you. So that's why we could just do less to a gzip file and we saw it okay. But you can, when you're using gzip files in pipelines, it's very handy to use gzip to uncompress it into a stream. So here, gzip-c-d, the C means to read and write from standard input to standard output, whichever direction makes sense, depending on which way you're going. And the D means decompress. And so here, what this is going to do is leave this gzip file on disk. It starts spitting out to standard output an uncompressed copy of it. So this is a very convenient way to start using gzip files in pipelines, because this lets the gzip stay gzipped on disk, but lets us use the uncompressed form in a pipeline and so on. So if I pipe this to less, you'll see that that's actually the same thing, which is basically what less is doing inside when you don't do it that way. But the fact that we have this now in uncompressed form, starting a pipeline, means we can now run other commands with the uncompressed output of this thing. So I could look at the first 20 lines, Maybe I want the first 20, first 5 reads. I could use some shell arithmetic there to make that easier. 5 times 4, 4 lines per read, 5 reads. There's the first 5 reads. Maybe I don't want all the garbage. Maybe I just want the bases themselves. How can I do that? Well, for most of these Unix text utilities are kind of a 1 line at a time kind of thing, but this is 4 lines in a group. So one thing that would be convenient to do is actually to rewrite this as sort of a 1 line per read format. So how can we do that? Well, here's a little magic to do that. Paste is a program that takes a bunch of files as arguments, and then the output of line 1 is the first line of the first file tab, first line of the second file tab, first line of the third file tab, and so on. So let me make a little temporary file here. This is A1, A2, A3. Here's B, B1, B2, B3. There's C, C1, C2, C3. Okay, so there's A, B, there's C. If I do paste ABC, you get this. Okay, and those are tabs. I'll change tab to @ sign. Okay, we'll get tab. So here we do this little gzip here. Where am I? Here. Paste dash 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 dash. Now what is that? There's no file dash anywhere. Well, a lot of Unix utilities use dash to mean standard input, which is basically what's coming out of this pipe. But then I've used it 4 times. Well, paste doesn't care. It's still gonna, when it goes to compose the first output line, oh, I need to grab the next line from this file, and the next line from this file, and then the next line from this file, and the next line from this file. Well, it's all the same file. It's all the standard input, which is what's coming out of the gzip. And that'll basically do exactly what we want. Take the lines in groups of 4 and turn them into columns 1 to 4. So there we go. So now I've got, there's line 1, oops, and then a tab, and then there's line 2, and then a tab, and then there's line 3, and then a tab, and there's line 4. Well, now I could cut to default is to use tabs as the separator. So I could tell cut, give me column 2. Okay, bam. All right, now I've got just the reads. Okay. All right. So I mean, this gives you a little flavor now about maybe I want to like write that. Maybe I'm doing some analysis. Let me make this not be the entire problem. So there's the first 100 lines, maybe just less, so you can see it on screen. There's the first 10 lines. So, I could then maybe I want to save that in a file because maybe that's interesting to me, and that's the next step of my analysis or something. This doesn't particularly make sense here, but then I could actually, whatever. I could actually use gzip to compress that into an output file. And so now I have, so there's an example of sort of like this idea of like streaming computation with intermediates not stored and gzip on both ends. So we leave a file gzipped on disk to get sort of an uncompressed transitory streaming copy, process it in the way we want to process it, and then we can take the output of that and gzip it lie and then dump that to disk. And so that's sort of a gzip to gzip computation where we're doing some stuff in the middle. This is a very kind of typical pattern that you can do, and so on. Okay, so this was a bunch of examples here we were working through. Here, another example I did on the slides here was to de-SRAify the names, right? So sort of like, it's kind of stupid, like, where is it? Where was the last? So if you've looked at reads from the cores, sequencing cores, you'll probably see something more like this as the names, without this and without this, and then just the plus sign here. And that's more closer to what the Illumina sequencers produce directly as their output. So this is sort of an SRA, an NCBI SRA format FASTQ file. Maybe we want to have an Illumina standard type FASTQ file. Well, what would that involve? Well, we'd have to pluck out this this part of the name and put that first, and then on the third line we'd have to get rid of this stuff or whatever. How might we do that? Well, okay, unzip it, paste cut character 2 to the end. That gets rid of the @ sign on the front of the name, and then we might use awk. Awk is something maybe you saw in workshop 1. That's a little kind of text processor that can do somewhat more fancy things than some of the other utilities, because it takes a little program that you can give it. Usually awk programs are very short. If you need to know more about awk, there's a link there to the awk page. This tells awk that we're using tabs to separate what awk calls the columns. And here we further separate column 1, that's $1, by spaces, and put that into an awk array called name. That's what's going to take apart. Here's my terminal. So then this will become the first part of the name. This will become the second part of the name. This will become the third part of the name. And then we're going to print something back out. And there's the @ sign that starts the name. And then we're going to do a string space a string, which will be the second part of the name followed by the first part of the name. So that basically will put this part space and then this part, or don't have this part. If you don't want the $1, you could just take that out. And then we do a new line. And then this is the sequence on column 2. We spit that out, and then a new line, and then a single plus sign, and then a new line. That's the third, what used to be the third line. And then we do the per base quality scores. That's the fourth column and whatever. And so if we run that, I get the start and ending right here. There we go. And now we have a FASTQ file that looks a lot more like an Illumina FASTQ file. So maybe that's something that we then want to gzip and rewrite out, and that's sort of the dsrified version of this thing. Now, if you run this, it's going to take a very long time. I think the estimate is like 10 hours. I'll tell you how I know that in a second. So if I just run it like this, you're going to get no output for 10 hours, except our session is going to die in 3 hours from now or so, because we asked for 3.5 hours. This isn't going to work. So Control C to stop that. One thing you can do in the shell is if you put an ampersand at the end of something, it puts the job in the background. It starts the job, sets it running, but then immediately gives you a shell prompt. So this is called a background job. We now have that thing running. If I ask the machine what processes are running as myself on the current node with this PS command, you'll see that I've got this SSH session is of how the network communication is happening back to the login node. This bash is my login bash. That's the bash I'm talking to right here. This PS is the PS command that I'm running right now to see the list of processes. And then you see the pieces of my pipeline, right? So then you see that here's the gzip cd as a process, the paste as a process, the cut as a process, and the awk as a process. That's the pipeline. It's these 4 Unix processes cooperating. And in fact, they're running simultaneously. So here again is an illustration of what I mean. When you run shell pipelines, you get kind of automatic parallelization, because each of those pieces of the pipeline is an independent process in Unix, and they're all able to run at the same time. So you get the multiprocessing kind of for free. Now, it still doesn't tell you how fast this is going, and at least we can look around right now. Let me get rid of some of these temporary files that we made. So here's the file that is being made. This is as it comes out. So I can look here, and you can see it's dribbling out, right? So you can see the file size growing. Now we don't know how big it's going to be on the end. I mean, first guess it's probably going to be more or less similar to the input file size, because it hasn't done really that much. I mean, it looks shorter because we threw away a lot of stuff, but gzip is able to compress those repeated strings very well. So first guess would be actually the file size that we're aiming at is something more like this. And you can see we've been waiting a minute or two here, and we haven't even, we got to get the digits out to here. It's going to be a while. But another kind of cute trick in pipelines is to use a program called Pipe Viewer. So that's not on Hoffman 2 by default, but if you run this command, you can use my copy. So that just makes it so that now you can use the PV command. All right. So that changes your path and so on to use this command I've already compiled and put in MySpace, since it's not part of Hoffman. But PV is kind of cool because you can just throw it anywhere in a pipeline, and it will take the input. If you're receiving input, it just passes it along. Or if you use it to PV with a file name, it starts producing that as output, and then you can put a pipe to the right. Or if you have it stuff to the left, it takes input, and so on. So you can just sort of insert it anywhere in a pipeline, but then it gives you a cool text progress bar about how far it's going. So here's the input file, and I'll give that to PV. And all PV is going to do is start spitting that file out in binary. Actually, it doesn't ungzip it or anything. It just passes it through literally. But what it does is it says, oh, I know how big this file is, because you can look on disk and say, this is how big the file is. So I know where I am in it as I feed it to the guy to the right of me. And I'll give you a little text progress bar for that. So then the rest of this pipeline is exactly the same as we already had, except I don't need to put the file name for gzip anymore because gzip will just read from the standard input if it doesn't get a file name. So essentially all I did was delete the file name from the gzip place and put pv, that file name, on the front here. And then you'll see the little coolness we get with that. Okay, so now you see this little cool progress bar that says we're 6 megabytes into the gzip file. We're 10 seconds into the computation. We're proceeding at about 740 kilobytes a second. We are 0% done. The ETA is 14.5 hours. So this is cool. You can kind of throw this into your pipelines and kind of get some progress information. I mean, for the interactive case. Right. So then we get to talking about shell loops, which we'll see in a second. But so if I have a bunch of files and I want to do some operation on all of them, you can do things like this. So for X in stuff means it'll do the body, which is until the done, which was here. Do this once, first time around with X equal to A, bam. Do then X equals B, bam. Then X equals C, bam. So put your commands here that you want to run repeatedly, parameterize them with a shell variable, wrap a for loop around the outside, and now the commands will repeat across that list with there. Suppose you actually had to do a computation that involved, you didn't just want to write to the screen, but you had to want to transform each file to make a new output file or something. So, you know, the word count command will tell you how many lines, how many characters, words, and lines are in a file or something. I just need a command right now that just produces some output. So if you wanted to do that, you would do something like this. So you could do word count, take input from X, write input to X.wc. Okay. And now you'll see we have A.wc, B.c, CWC. So that actually gives you an idea of more how you do something more useful, right? You know, just replace WC with something that does something more interesting. But that actually also brings up the fact of how to actually parallelize things on a single machine. I could just replace this semicolon with an ampersand. And now I just ran those 3 jobs. The ampersand puts the job in the background. And so the next iteration of the for loop will start even before the WC of the first one finishes. So we've actually, now WC is too fast, right? So these were done 0.001 second after I hit the return. So I need a command that's a little slower. So all right, I'm just doing random stuff. So sleep is a command that just does nothing. Sleep takes an integer number of seconds. Maybe you can do a floating point, I don't know. And then when it's done, it just exits. It just wastes time. So let me do this just so that I have a command that doesn't finish instantaneously. So bam. Now I've got 3 sleeps running, and you see they're actually all running together, right? So this is actually a way that on a single node, or your local machine or single node in Hoffman, in an interactive session, you can run things. This is how you do. Don't do 1,000 jobs this way. But if you've got 10 or something, right, this is possible, right? So this is very, very easy, right? All we had to do is basically put an ampersand in there, and we're getting now kind of like, it's almost like a little job system, right? Like Hoffman, but I mean, it's a shitty job system, but it'll get the job done for small cases. So this is, it's all about choosing the right level, right? So you've got a small bunch of things to do at once. Yeah, this is a great way to do it. Very, very easy. Maybe it's been a minute. I guess they're still running. If you do need to stop them, so you can do various things. So like FG, they're all done. All right, okay, I got to start them again. Maybe, okay, that's 10 minutes now. Okay, so if you wanted to, if these were producing output to the screen or something, you can use FG to bring them into the foreground. So now job 1 is actually connected back to the screen keyboard. Control Z puts it back into the stop state. So now you see it's stopped. BG, I could put it back into the background. Now it's running again. Kill is how you get rid of them. So I could kill job 2. Okay, so now job 2 is gone. How do you kill all of them? Jobs-P will give you the ID numbers there's these percent numbers, but also process IDs. That's what the jobs.p kill takes either one. So if you really get stuck, you essentially need to kill, the 6578 here and 6580. That's not so convenient, but you can use another shell feature called process substitution. So one of the things you can do in the shell is if you use, we learn shell arithmetic is like this. With only one parenthesis, it's called process substitution. What this does is run the things in between the parentheses as a command, take the standard output of that and put that into the shell command line and then run that command. So here the date expanded into this and then the echo just printed it out. But if you reach a situation here, let's see, where's my launcher? Okay, here's another style for loops, more like traditional computer code. That makes X go from 1 to 20. Okay, so this is run this at the start of the loop. This is how you tell if you're done with all the iterations, and this is how you move to the next iteration. Well, now I've started a lot more of these and so on. Now it would be a real pain to kill these guys. So I can take the jobs-b that gives me all the ID numbers and pass that. Here, I'll put an echo in front so you'll see what that expands into. Right? So that expands into this. But now I'll just get rid of the echo. So, okay. All right. So these are all good illustrations of how to use shell stuff to get stuff done. All right. We should take a short break because it's been an hour, and that's okay. We're not getting as fast as I thought, but actually we're kind of pulling in things that I thought we're going to cover later, and we're covering them dynamically, kind of like at the front here. So we're going to be able to go faster later through some parts because we've already seen a bunch of things. It's sort of all happening all at once. It's sort of everything everywhere all at once. But anyway, let's take a little break. I don't know, 5 minutes, whatever. If you have to go to the bathroom, go to the bathroom. If you have any questions, ask questions. There you go. Ask you like for some. Just got any bottles of water. So, units of operating system with the different copies. Things like individual copies that exist, space, Excel, when we have a pipeline like A, pipe B, pipe B, pipe B, A, B, C, D are all one individual copy. They have, Unix also has the idea of file number 1, 2, 3, 4, 5, 7. Each of those can be connected to a file or not. Unix, everything is a file. Ordinary files are files, register files, network files, keyboards are files, series are files, whatever. A file is basically something that you can read from and or write to. Some files support something around random. Keyboard can only go and print them. And the way the pipeline works is that there are 3 standard file descriptors. 1, 2, 0, 1, and 2. 0 is standard input, 1 is standard output, and 3 is standard output. And the convention for programs is when you use input, you have to read the file. For things like default utilities that you mentioned, if you need input, you read the 0. The right output, ordinary output, you write the 1, error. And the way the pipelines work is that you have A plus B, which is really say connect A standard out to B standard. Through a little tiny buffer. And so A starts to write output, it goes into the buffer until B is ready to read it. And if B is really slow, A will fill up the buffer, and then A will have to stop. Because now A will try to write into the buffer, but the buffer is full. So basically, A and B's execution is a little tied together because if they get really out of sync, one's going to have to wait. But if they're both kind of proceeding at comparable pace or whatever, then yeah, they're both running. You also basically, I mean, modern speed is probably how the old days our pipelines are only having a few. All so A and B are probably running, simultaneously. Even with one core, you can run simultaneously, right? If you run a slice of A for a millisecond, and then switch to B and B for a millisecond. So you can have more, in the old days you had one core, these days you have 32 cores. But you can still run more processes than you have cores. But yeah, so you still call that simultaneous. But yeah, probably A type B, probably, unless A and B get really out of sync in terms of doing one and one, yeah, they're running. And what does the ampersand do? Nothing, basically. It just controls whether the shell waits for the processes to start to finish before giving you another cell time. Okay. If you don't print the ampersand, the shell is just sitting there doing nothing, waiting for its children. If you do put the ampersand there, the shell immediately returns. It's got an ear out, so this is when the children finish, which is how you do the static. But in terms of the actual computation, you have to put it there. It's just does the shell wait for them to finish? Okay. All right. Where were we? Yeah, basic job management. That was actually here, which we already kind of went through. And we will see more of it as we go on. Yeah, anyway, we saw that this let me just clean up. We don't need all this demo stuff. This one. There we go. Is this thing still running? No. Okay, because this wasn't even finished. Okay. All right, I think we're back to clean, ready to proceed. Okay. Right. Great. Okay. Now for long running computations, and sometimes you'll see this kind of pattern, which is helpful. When you have a file, when you have a long running computation, like say that SRA one that we just deleted, right? So like this we put it in the back. I'll just start in the background again. I mean, here, this is the background in the background. There it goes. So this is being generated, but it's not done yet, right? Now we know it's not done yet because we're the one that started this thing and so on, and we have this running job and so on. But it's not obvious whether, like from the file name, whether this thing is done yet or not. I mean, it looks like a .fastq.gz. It's got a size or whatever. If we started gunzipping it, even though it's not done yet, gzip is a streaming computation. So the beginning of this thing, gzip will be able to uncompress. I mean, eventually, if I go far enough, I'll reach the live end, and then gzip will say, yeah, I don't know, it looks truncated. I'm expecting the way the compressed format works, this ending isn't possible. Whoa. We lost the project. Okay. But suppose this was something that we went to sleep and woke up in the morning and whatever. I mean, or you come back, maybe your Hoffman session disconnected, your internet disconnected like Time Warner always does in the night or something, and you lost your network connection. It's like, is this thing done or not? Well, you don't really know unless you have other logging or something like that or whatever. So one pattern that sort of makes that a little more obvious when something's done or not is to actually not name make the file name something that tells you it's still in progress, and have the very last step of the computation rename it from in progress to not in progress anymore. So that way, if the file name says the in progress tag is missing, it kind of reached the end. So I'll show you kind of this very common pattern like this. So I'm going to get rid of this thing again because its purpose right now is done. Okay. Okay. So here's the pattern I'm talking about. So this is just a stupid example, right? So I'm going to print the date right now, wait a minute, and then print the date again. parentheses make what's called a subshell. Basically, a new shell is created. It'll run all the stuff between the parentheses, but things like redirections, which we can put outside the parentheses, then apply to this whole subshell. So all these things, all these things will redirect into the same place. Double ampersand is like semicolon in that it runs the thing to the left and then the thing to the right. But it's a little trickier than that. We haven't talked about it yet, but in Unix, every process produces an exit status, which is an integer between 0 and 255, with the convention that 0 means success and non-zero means failure. And all good programs, which all the standard utilities are, and all the shell stuff is, if there's, if you tried to cat a file and it doesn't exist, cat's going to return a non-zero exit status. If you gzip and gzip runs out of disk space, like you actually fill up the disk completely or something, gzip will return a non-zero exit status. And so every program is sort of declaring to the shell when it finishes whether it kind of reached success or not by its own definition. And what the ampersand, double ampersand done says, only run the thing to the right if the thing to the left succeeded, okay? Whereas semicolon doesn't care. Semicolon says, run the thing to the left, don't care. When that's finished, run the thing to the right. The double ampersand says, run the thing to the left, only if that succeeded, continue on to the thing on the right. So what we do here is, we do the redirection, we want to produce this file demo.txt, but we start during the computation, we call it demo in progress txt. But then when the computation finishes, we rename demo in progress to demo. That way, since renaming is an atomic operation on the file system, if we come back to this thing hours later or next day or whatever, and we still see demo in progress txt, we know it didn't make it to the end. If we see demo.txt, we know that our commands reached the end. So that allows us to turn the indication of whether everything kind of made it to the end or not into just looking at the file name, which is convenient. Here's how that would look. So I'm going to put this in the background so we can poke around while this runs. So it's running in the background, okay? And you will see the demo in progress txt. And because of that sleep, you're going to get the first date, but not the second date yet, right? So if we wait a minute, we're going to get the second date. All right, so let's not let it wait a minute, okay? So suppose I kill it now, okay? So now it's dead. Well, now it's gonna stay in progress txt and it's gonna have the one line, but now it's not complete. Okay, so let me get rid of it. Now let me change the time to maybe 10 seconds so we don't have to wait as long. Okay, now we'll let it go to completion. Okay, so there it is. There it is, we can look at it. If I catch it fast enough, there's only one line. And I'm waiting here. Let's see. There it goes. Done. Okay. And now if you look, now it's got both and it's named correctly. So that's the indication that this is now a complete file. So you'll see this a lot in pipelines, especially ones that have commands inside that run a long time or produce output kind of in whatever. Alignment is kind of one of them or something, because you're going to have a big FASTQ input file and a big BAM file in the output. So good idea to have the aligner produce this output in progress BAM, and then only if that alignment kind of reached the end and is successful, do you rename the in progress BAM to BAM. That way you know whether your alignment's finished or not. Okay. Now, so these files are still too big to use as a demo for today. Okay. And we don't really need them this big to show you how it would work with all the parts and stuff like this. I can just cut them into smaller parts to still get a large number of parts and show you how to deal with a large number of parts. And then we don't have to wait so long for the whole thing is because we're doing this live. So we're going to cut them down. So first of all, how big are they? So here's a little pipeline to count how many lines are in them, which I've already run because it takes like an hour or something, because as it turns out, uncompressed, these things are over 100 gigabytes each. And they have over 1 billion lines each. Like the first one has 1.8 billion lines, and the next one has 1.4 billion lines. And then if you divide by 4, you find out how many reads there are. That's like this and like this. So this is actually a long-running computation. So it's a perfect example of something to put in the background. I've already got this run with .wc as the file name. So we're going to run this live with .C because I don't want it to erase the ones that I already have there. But what this does is that that these are file name patterns, right? So like with the star and stuff like this, you can have that expand to the shell expands that to the list of file names that match the pattern. Okay, so there's my two files that I want to process. We already did the for loop for how you kind of do a command against multiple files. And so this is that same kind of loop. And what we're doing is we're unzipping it and passing it to WC and then writing WC's output to a file name. Now, a very common problem when you have sort of this kind of pattern of like do the same thing to a bunch of files, but then you need the output file names to kind of change along with the input file names. But we only have a shell variable for the input file name. So how can we construct the output file name for the input file name? Well, the easiest thing to do, I suppose, would be to just tack on some suffix on the end, right? So we could have .fastq.gz.wc, but oftentimes you don't want to do that. I mean, eventually you just end up with so many dots and it's kind of crazy. What we kind of want to do is maybe strip off the suffix and put a new suffix on the end. So maybe when we want to replace the .fastq.gz with .wc. So the shell variable expansion has all kinds of little operators that you can put in it. There's really a lot. There's like 30 or something. But one of the most important ones to know is a %sign. And that means look for this pattern on the end of the thing and get rid of it. If it's not there, it does nothing. But if it's there, it gets rid of it. So we take X, kill the .gz on the end, and then we put a .wc on the end of our own. Okay, so if I, here let me. So let me, instead of running this, there you go. We're just going to do that little part. So we're just going to, okay, so we're going to do this, but we're just going to print out the result of that little expansion there. Okay, there. So what do you see is the .fastq.wc. If I wanted to get rid of the fastqs too, I could do that. I could put .fastq here, and now that would match that suffix. So now we just get the .wc there. So this is a very common pattern that you'll see in processing files, is that you have a shell loop over some input files. So you do a 4x over inputs, but then somewhere you got to have, I mean, I could have done this, a little kind of more verbosely like this. But maybe this would be a little clearer, but all right, that's the same thing. It's making an intermediate shell variable with that. But this is a very common pattern. You're doing some commands in a loop with input files. The loop is over the input files, but you need to construct the output file names, and you do something like this. You take the input file name and you either pull out pieces of it, and if it's like you pull out a sample name or something, you get rid of the suffix or something, and then you tack on something on the end that indicates what kind of computation you just did on it that this output file is recording. Anyway, very, very, very common pattern to see there. So here we just want to count the size of these files, and because these files are so large and it takes a while, we don't do it interactively. We just put it in the background and let them start grinding away. So that's this. I already have the outputs that will eventually come out of those, but it's like an hour. So actually, I started it twice. So there we go. Let's just delete it. So I'm not going to wait for those because we've already got the results in the .wc files. So this is the number of lines, the number of words, which we don't care about, and the number of characters, which is maybe you care about. It's the uncompressed file size. If you unzip these files, that's how big they would be. Right? If you do this, the file that you get .fastq at the end is going to be this size, which is 147 gigabytes almost. Anyway, so yeah, so these are big. Here's how many lines they are. Remember, fastq is a 4-line format. So if we wanted to get the number of reads, we should divide by 4. Well, here it's very easy. They're so small. We just put this into a calculator and we could just divide it by 4, and there you go. That's the number of reads in the first file. But maybe you have 100 samples or something, and you don't want to do that. So here's another little shell example of how you would just automate the extraction of that first column, and then divide it by 4 and attach the file name on the side. And this will introduce shell functions. Now, shell functions are basically little bits of shell code, but they can take their own parameters. So the $, you may know when you write a shell script, right? We haven't talked about shell scripts yet, but I think you saw those in workshop 1. You put the shebang line here, right? And then let's see here. The arguments are to the shell script are in $1, $2, and so on. I use the braces because if you ever get to the point, you may have seen these as $1, $2. Braces are always a good idea because otherwise you're going to get surprised, because $10 is really interpreted as $1 followed by a 0, and so on. You really want $10 in brackets. So I always use the brackets just so you don't have to think about it. So maybe we just do something like this. right? And then you know you have to make them executable, right? There are various ways to do that, chmod or whatever, but you need to get this executable bit on the shell script, and then you can run the shell script. Well, I didn't give it any arguments, so all the arguments are empty strings. So empty string, and arithmetic understands unset and empty as 0. So that's that, but really you're supposed to do something more like this, right? So those are shell script, right? So they get their arguments as $1, $2, and so on. And shell functions are the same thing. They also get their arguments as $, $2, and so on. But you can wrap them up, curly braces, give them a name, and then you can call them from other bits of shell code. I'll explain the local in a second. But basically, this is supposed to take the output of WC, which is lines, words, and columns, and then produce lines divided by 4, right? So if I give you this thing like 400 and then the rest doesn't matter, it's not going to pay attention to those. It pulls out the first one, divides it by 4. So that's good. The local is just related to, do you want the L, W, and C to leak out outside the function or not? So by default, variables inside functions are operating in the shell environment outside the function. So if I took this L, W, and C out here, then I'm actually changing L, W, and C also out here, which maybe would interfere with something out here. Here it doesn't, but in general, you kind of want isolation between the inside of the function and the outside of the function. The local means that the variables L, W, and C only are their values. If they had values, they're saved. They can do whatever they want inside the function, and when the function quits, they're put back to the way they were. So this makes it so that the L, W, and C don't kind of interact with the outside the function world. Just kind of good practice. So then we do a little loop over our WC files, and then we cat the WC file, which is going to be something like this. We use the process substitution to take the output of that and stick that as the arguments into first arg4. Then we run that as a shell thing and use command substitution to take the output of that and put it into the shell variable nreads. And then we echo nreads tab, and then the file name with the .wc removed at the end and replace the .fastq.gz. And then we take the entire output of that for loop and redirect it into a little file. So what's the net effect of all that stuff? Now we have a little 2-column tab separated file. First column, number of reads. Second column, file name. Except it's a little wrong because due to the choices I've made, I've got the extensions wrong. So I think there's a fast queue missing here. Right? So I need to remove more. I got the fast queue came out twice in the output, so that can't be right. So yeah, that's better. That's what was intended. Okay, so you can imagine now if we had hundreds of samples or whatever, that's great. We don't have to manually do things anyway. Basically, anytime you have a task, if you're sitting at the shell and you're doing something repetitively, there's a way to automate it. Basically, that's the lesson. Sorry? Yeah, okay, so I don't know. We'll get to a point where that is explained. We could do this now. This is the explanation for why. So here, outside A is 5, and then there's all these echoes that just tell you what the status is at the moment. Then we call this function global bump A, and then we print out A again. And bump A, the only thing inside here of any importance is this, which just increments A by 1. And the question is the A, so what's the value inside going to look like before and after, and what's the value outside going to look like before and after? And so, and notice this does not have a local. So in this example, starts off at 5 outside, then we go inside the function. When the function starts, it's still 5. The function increments it by 1. But because the function is really twiddling the global A, when we go back to the outside, A is now 6 too. Contrast that to the situation with local. I put a local A here. Local A makes A unset, and its value is now private to the function. So now, when I run the same thing, but basically all I've done is inserted local. So now, starts off as 5, we go inside, it starts 5, but then I hit the local, now it's unset. Then we do A equals A plus 1, but that's arithmetic with an unset value plus 1, and empties and unsets treated as 0. So 0 plus 1 is 1, so that's why A is now 1 inside. But now we return back to the outside, and now A is still 5. because the a inside the function is local and does not change any of the outside a. That's the illustration of what local versus not local does. A lot of times your shell functions are so close to the code that calls them, and it's really obvious that none of the variables inside the function are going to collide with anything outside, and it's not a big deal. But when you start getting a pile of shell functions together and you want to sort of like get a little library of them going and you start calling them more randomly, then you sort of kind of want to have some protection that the insides and the outsides aren't going to interact in weird ways. And then local is a good idea. Okay, so where were we? Anyway, yeah, so I mean, this is pointless to automate this for this size problem. You know, the idea is, you know, to show you how things scale. Okay, blah, blah, blah. Okay, so yes, so we know how big they are now, right? Okay, so there's like 450 million reads and 350 million reads. I told you at the beginning, but this is how you verify and get the exact number. So yeah, these are too big to do the live demos with. And so while a real BS alignment split into pieces run on Hoffman job, maybe a typical size for the piece would be something like 10, 20, 30 million for a piece. We're going to cut both of these files down to the first 10.1 million reads. I could do 10 million reads, but I do the 10.1 so that we get a piece at the end that's not full size. Because in general, you know, files have random number of sizes, and you cut them into fixed size pieces. Usually the last piece is partial. So I want to make sure that you see the partial case. So 10.1 million reads, and we'll break them into 500K pieces. Okay, that'll make 21 pieces for each file, because 10 million into half a million is 20, and then the partial 100K at the end is the 21st piece. So sample A will be 21, sample B will be 21, it'll be 42 pieces together, and that will be our demo is 42 parts. So we need to now construct these partial files and so on. So how can we do that? Well, we can do it like this. The command we want to run is something like gzip cd, the input fastqgz, the big one. Then take the first, how many lines? 10 million plus 100,000 times 4, right? Because that's 4. It's a, you know, it's fastq. It should be 4 lines per read, and then this is a more, you know, this is really obvious that it's. I find if you just, you know, I could have just replaced this whole stupid thing with what, you know, 10, 0, 0, 0, 0, 0, 0, and then what? There's a 1 there, and then we got to multiply by 4. Basically, that's all it's going to be. Right? I mean, if you could do it, but so, you know, sort of like also you kind of see, maybe you appreciate the documentation aspect of sort of embedding the arithmetic in this directly as arithmetic. The intent is a little more clear. That'll give us the right prefix. Now, to really, I mean, if we were really doing this in a real biological application, you should not take the top of the file. You really want to do random sampling of the whole file. Which is, you need a fancier program to do, because you need to do something like reservoir sampling or something to do that with a streaming operation. But we're not using this data. We don't care. I just want to cut them down to a size that works. Again, we're not doing this for the biology. We're doing this for the study of the computation. So this will be fine. We'll just take the top of the file. Then we re-gzip it. Now we need to write a new fastq file output. So let's simplify the file names. Remember, percent deletes the suffix if it matches the pattern. The pattern is double dash followed by anything. So we will look, if you look at these input file names, wherever we have them on screen somewhere, here, the double dash occurs here. So we're going to basically match double dash star, which is going to be all this junk. And we're basically just going to keep the sample name, which is this thing at the front. So this part is basically the sample name, and then we're going to add dash head to remind us this is the 10.1 million head of it, read head of it, .fastq.gz. We'll do them both in parallel and go. All right, so let's get this going. Bam. Okay, so they're the 2 running. If I catch it fast enough, we can check out what processes are running at the Unix level. You see, I've got 2 gzip compressors, 2 gzip decompressors, 2 heads running. So we've got 6 processes in flight, all working together at once here. This won't take that long because, you know, these are the files that are growing. All right, I mean, how long is this going to take? We could kind of, you know, if we're getting, oh, well, we're done. Okay, that's about what was done. So there we go. We don't need this. Oh, okay. I was going to show you the watch command just to show you. So you saw how I was like hitting up arrow, enter, up arrow. I was doing this like manually or whatever. There's a program that does that too for you. So let me run that computation again. Okay, there we go. It's going to do the same thing again, overwrite the existing files. But I'm going to run this little watch command. What watch is going to do is it takes a shell command and runs it over and over again, like every 2 seconds by default, I think, or whatever. So basically, that's just going to do what I was just doing manually. Looks like the terminal window's a little messed up. I think there's this, like it's shifted by one character, like it's shifted by one character or something like this one. This one shouldn't be here. This one should be over here. And so I think this 4 is not really, or maybe it's this 2 on the front. It's not really there. But anyway, so you see that's just basically running the ls command over and over and over for us. And then the set-b, you may have noticed that when I was doing the background stuff that the shell doesn't tell you when a job is finished until you kind of run the next command. The shell is always waiting. It doesn't like tell you updates for your background or background commands until, like if you're sitting at a prompt, it will never interrupt your prompt. It'll let you type your business. When you hit enter, and before you get the next prompt, that's when it tells you about what's gone on for the stuff it hasn't told you yet. The set-b tells you, let me know right away. So I wasn't on the screen here, but basically, even though I haven't gone back to the original shell prompt, you'll see that I got these done messages here. That's what the set-b did. Is it said, tell me right away. Don't wait for me to do my thing, right? So I could show you that again. Like so, like here, wait 5 seconds in the background. Okay, 1, 2, 3, 4. Oh, because I still got the set-b on. Okay, so all right, so that's set-b, but the default is actually set+b. 1, 2, 3, 4, 5. Should be done. Should be done. No message, because it's going to wait until you give the next command to give you the usual updates. This is the intent here is that if it doesn't want to sort of like interrupt things that are going on. Like here, you're typing a command in this prompt. It doesn't want to like mess that up. Or if you've got a command producing output, it doesn't want to mess that up by inserting its text in the middle somewhere. So that's the rationale there. Or sometimes you want to turn that off. Anyway, yeah, there we go. Okay, so there's these guys, right? Oh yeah, I'm not going to bother to explain exactly how this one works. This is going to be sort of a monstrosity, but there are ways to make Pipe Viewer actually show you all the progress bars at the same time. Terminal had a little hiccup there, but that's, you can really kind of get fancy with this stuff if you get into it. So there we're actually seeing both of the flows at the same time. That here, the Pipe Viewer is actually measuring the size of the output. You don't want to measure the size of the input because we're not going to read all the input. But we know we're supposed to produce output files of a certain length, 40.4 million lines. So we tell Pipe Viewer, we put Pipe Viewer at the point where the output comes out. We tell Pipe Viewer measure the progress bars relative to 40.4 million, and so it's able to do this. Anyway, this is diminishing return. Anyway, there it is. Same thing. We just did the same thing 3 times. Plus anyway, okay. So how we could another thing you get in the habit of doing is every time you run one of these things, you got to check, did it actually work, right? Because making these commands isn't always straightforward. So the question is, did this do what I thought it did, right? So basic check, what's the size of the outputs? So they're about a gig each. How many reads are in each one of them? Well, we can use that little WC, gzip, WC, first argdiff4, blah, 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 echo, so on. We can use the thing we already developed to tell us how many reads are in each of these files, right? So here, I didn't do them in the background because they should be short enough that this doesn't take that long. So I didn't put the ampersand there and redirect the output to files. We're just going to get it out one at a time and run it serially, but come on. Didn't take that long. 10.1 million. Exactly right. On the other, well, okay, then the next one's going to come. Oh, yeah. There's also more, one of the computer science acronyms that's very common is Tim Towdy. There's more than one way to do it. Okay, always true in computing. So this is what we did because we already developed this kind of thing. But maybe a more literal thing would be something like this. We just do gzip cd paste 4, count the number of lines. This sort of divides it by 4 right here, right? Because we were doing this kind of thing already. So this would be just perfectly fine way to do it too, and it gives us the same answer. Okay, so I'll run the second way, and that can work while we're talking about this last thing. So we're at the point now where now we can make the pieces. Okay, let's actually make the pieces. How do we cut these, these big FASTQs into little FASTQs? Then we can worry about the problem of how we align the little FASTQs, but we've got to make the parts first. How do we make the parts? Well, the command you really wanted to know about is split, which is one of the basic utilities like grep and cat and all the rest. And that's its main function, is to split big files into small files. So how does that work? Do I hit Control C or do I wait 5 seconds? 4, 3, 2, 1. Yes, no? Oh. Sunk cost fallacy. Do you wait it out or give up? There you go. So man split. You can read through this, but the options that kind of are important are, well, let's do a little tiny. Yeah, we can do a little tiny demo. So what split's gonna, maybe I'll just explain it. Split is gonna take one input file, and then it's got to make a lot of output files, right? So we can't really name all the output files because that would be very messy. We'd have to know how many parts there were, and we'd have to construct all the output names anyway. So split does the job of naming the output files itself. How does it name the output files? You give it a prefix, and then it tacks on a part number at the end. And the part number can either be alphabetic, like AAA, AAB, AAC. I don't like that form. You can also make it do numeric suffixes, like 1, 2, 3, or usually with leading zeros. And it actually likes to count from 0. So more like 000, 001, 010, 011, and so on. So that's how the naming works. And then you have to say how big the pieces should be. That can either be measured in characters, doesn't make any sense for FASTQ files, or lines. That makes a lot of sense for FASTQ files. So if we want to cut the files into, we decided on what 500,000 pieces, that means 2 million lines, right? So we want to break into 2 million line pieces, and we can decide what names we want and so on. So basically the options that are relevant are this thing. This tells it how many digits you want to use. So in this case, we know we're going to need 42 parts, right? We already, that's kind of the point of actually pre-flighting the rest of this, right? So we know how many reads there are. We know our pipe, we know our size of pieces. We know, well, sorry, that was before we cut it down. We cut them down to this size, and we know the piece size. And this together already implies the maximum number of pieces any single sample needs is 21. They both need exactly. So 2 digits for the part numbers will suffice. And so it allows, and that is in fact the default. But in general, if you're doing 1,000 pieces, you're going to need suffix like 4, right? So, so anyway, so here's where you tell how many digits to put on the end. If it's not enough, you're in trouble. But if it's too long, I suppose that's not much of a problem. But I'll just set it to exactly what we need in this case, which is 2. And this is pieces by bytes. That doesn't make sense for us. This is break at a line, but still measure the pieces by byte size. Let's not do that either. Numeric suffixes, yes, we want this because I like the digit form. And in fact, I don't even pay attention to this. The man page actually tells you if you want it to start counting from 1, the default is 0. Actually, you can say numeric suffixes equals 1. Well, that would probably been good to pay attention to man page because we're going to have a little script we had to write to rename these things to count from 1, because that's nicer. But I now see that there's actually a command line option for that. So there you go. Paste to read the man page. And lines. This is the one we want. This is how many lines. This is max number of lines per part. So parts will always be the size. The last part may be partial. And then what else? That's basically the end. Okay, so blah, blah, blah. Okay, so let's put it together. So we read the man page, or we ran it a little more detailed than last time when I put the slides together, but okay. So we're going to make it more clear where this dependency on the number of digits in the part number is here by using an intermediate shell variables here. So we're going to be using 2-digit suffixes for the part number, 00 to 99 if you count from 0, 01 to 99 if you count from 1. Both are completely sufficient, 21 parts. We're going to loop over our input files. There's only 2 of them. These are now the 10.1 million cut down input files. So command is split. We tell split how long we want the suffix to be. We activate numeric suffixes. We tell the number of lines we want the parts to be, which is 500,000 times 4, because we need FASTQ as a 4-line per read format. This is now the input file. Now I've used another interesting piece of shell syntax, less than parens, which I'll explain in a second. But essentially the input file we want to be the FASTQ GZ, but we do not want split to cut the binary gzip file into pieces by carriage return characters. That doesn't make any sense at all. Split needs to operate on the uncompressed form. So the gzip cd x kind of makes sense, but how do we use that as a file name? Well, that's what the less than parens mean. It's a redirection like less is. You can we've used that a couple times, right? We must have. Yeah. So yeah, we've been using less to redirect input a couple times. Well, that's not a redirection there, but these are. So you know that you can redirect input with the less than, but normally after less than comes a file name. What we want instead is we want a shell pipeline, and we want the output of the shell pipeline to be treated as a file name. You don't have to worry about the magic inside that makes that happen. It's basically what's called a named pipe, or it's actually even doable with just file descriptor management, but it doesn't really matter how it works inside. But basically, the shell will make what looks like a file name and give it to the program, but when the program opens that file name, it's really connected to the output of this pipeline. And it's not done through temporary files on disk or anything. It's done through a pipe, the same way that pipelines are done. So it's a streaming 0 RAM kind of thing. So basically, split is going to split the input to split is essentially the uncompressed form of the file. The suffix for split is this thing, which is the input file name delete.fastq.gz and add -part, and then split will handle the suffixing of the parts, the part numbers after this. And then we'll do the 2 splits in parallel in this loop. Now, I think the reason why I didn't know about you can tell this thing to count from 1 instead of from 0. And actually, I see that there's even another option that split says, which says that you have this additional suffix so that after it does prefix part number, you can then have something else go after that. Like we're going to need .fastq at the end of those. I think that's because I grew up with an earlier form of split. This is GNU split. Most of Linux uses the GNU set of userland utilities. Some of the older stuff like BSD and so on, I don't think the split was as featureful, and so that's probably why I didn't think of it. But anyway, we're going to go with this, and then if we can fix it up later, not too hard. So anyway, so let's run this now. Okay, so bam. Okay, so those are the 2 jobs that are running. Do I have one of the PSs around? Percent, you may have, I think in workshop 1, we do tell you a little bit about the history command, which the shell is keeping track of the last, you can set how many, but in my case, it's about 1,000 commands, the last commands I've done. And history shows you the complete list. You can also put exclamation point, which is called history expansion, and pull back any of these commands from the history, like 1203 runs man split again, and so on. I can also grep my history, like that's how I was able to find, show me all the places I've used the greater than sign lately, stuff like that. Anyway, the reason I brought that up is because what did I do? Oh, I wanted to see the PS, so I just did exclamation point PS to pull back this PS command that I had most recently run. It looks for the most recent line that starts with that, or has it inside. Okay, anyway, I think those are done. Those actually were pretty fast. So what you see some water here. So here we go. So that's the big guy. We weren't splitting that. This is the cut down version of him, which is here. He has now been split into these, right? They've been numbered from 0. You do have 2 digits. The last one is going to be partial, all is expected. Okay, that's all looking fine. The other one was here. He is here. Now, these, of course, will be uncompressed because split has as input the uncompressed thing, but split, it's not really possible to wire gzip into the output because split is the thing opening and closing bunches of files and so on. We can't kind of insert a pipeline into the middle of that process that's internal to split. So these are, of course, going to be uncompressed outputs because split was an uncompressed to uncompressed thing. It has no awareness of compression or anything. So each of these parts is an uncompressed thingy. So we should name them appropriately. They deserve a .fastq extension then. We should probably gzip them to save disk space, because especially if you were having a real project, disk space becomes a serious concern. Like you could easily end up with a project that needs a terabyte of disk space for 1 copy of the reads, but then you've got sort of the inputs, the parted out copies, the BAM files, and then maybe you've got to merge the BAM files to create samples. You may have 4 copies, essentially, of things floating around at a time. You can easily be into the 3 or 4 terabytes of disk space with a big project. And so gzipping, and gzipping is very, very valuable on FASTQ files and SAM files and stuff like that. So can easily save 50% or even folds of space. And so you really want to keep things gzipped as much as possible. So we should put .fastq on the ends of these. We should gzip them. And then I don't like them numbering from 0. I like them to start from 1. That would have been easier to do by having the right option to split here with this numeric suffixes equals 1 or whatever, but too late now. So we'll have to fix it up outside how to do that. So we should probably check that these are kind of complete as well. So we could do that with a loop, sample 1, sample 2, word count, lines, all the part numbers. The word count, when given multiple files, will give you 1 line per file, but also a total line. Very convenient. So you'll see here, we have the right number of parts, all of them 2 million lines, 500K times 4. The last part, that should be 400,000 because that's the 100 by K extra. In total, 40.4 million lines, 10.1 million reads. They both had the right size. So that looks like the split did exactly the right thing. All right, let's take care of these 2 little problems. Add the extensions and renumber them plus shift by +1. This is really more of an excuse just to show you some more stuff about manipulating strings inside the shell. So there's going to be this function magic. Let me show you it operate, and then we'll talk a little bit about how it works. So here's this little magic shell function. And we're going to make it do the job by just doing this. So here's the ls, the situation. This is our shell script. It takes 2 arguments at the front, which is the number of digits in the part numbers, 2, the extension that you want to add to the end of the files, and then the names of all the files you want to process. Okay, which is all the srx star head part whatever's. Okay, so there, and you see it did the renumbering. So it took this one, renamed it to part 21, and put .fastq on the end. Now you notice a couple things here. It did them in reverse order from high part numbers to low part numbers. Can you tell me why it would be a bad idea to do it the other way around? Yeah, because if you try to do them in the forward direction, what would be the first renumbering? Part 0 to part 1, but there's already a part 1. Now you're in trouble. You got to do them backwards so that 20 goes to 21, which doesn't exist yet, making a hole at 20 so that 19 can go to 20 and so on. And that's why we do this backwards. The backwards is handled. The shell actually guarantees that when you expand file names with these shell patterns, they always come out sorted in dictionary order. So because of the leading zeros in the part numbers, these will become the numerical order ascending forward, which means that inside the shell script, we actually got to process them in reverse order that they come into the shell script, which is what this for loop will do it. Instead of running 1 to end x plus plus, it goes from n to 1 minus minus. That's that business. The rest of it is just pulling out the right pieces of the file name and junk. So here's the insides of this script. So first we got to digest the arguments. The first argument, remember, is how many digits in the part number. The next argument is the suffix you want to add to the end of the file names. And then here's a little kind of convenient thing. The shift thing in shell deletes that number of arguments and moves the rest forward. So if I get rid, because I just kind of captured this one and captured this one, then I'll get rid of them. And that means that all the arguments that are left are the file names that I should process. It kind of just simplifies the accounting and the rest of this. I don't have to start counting from 3 going to the end and so on. Now it's just 1 to n. So now I need to do, instead of, normally I could do a loop like, we've done this loop like for x, and you haven't seen this yet, but $@ in double quotation marks expands to all of the arguments. So if you have a shell script and you want to process all of the arguments in turn with their file names or something, you do this kind of pattern. That doesn't work here, unfortunately, because we need to process them backwards from right to left because of that business of overwriting files if we try to go to forward order. So we have to kind of do this in a stupid way. We have to, instead of using x to directly be the file name, we have to do x to say which number it is. So if there's 10 file names, x needs to go 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Then we'll convert the x equals 5 to the 5th positional parameter down here in a second. That's okay. So anyway, $number sign is how many arguments there are. So I don't have do I have a shell script around right now? No. So let's just say, let's just do something like this. Okay. Do I have it? Oh yeah. That's not that lovely shell. I think it's because the colon changes the meaning of that thing. Okay, let's just do it this way. So here's a little script. Oh, no. I'm missing a double quote mark. There is the problem. So there I ran it with no arguments. $ number is 0. There's with 1 argument. $ number is 1. There's 2 arguments on both of them and so on. You see the @ there is expanding to all the arguments. The $ number is expanding to the number of the arguments. So this is how many arguments are left after we took off the first 2. We keep doing this until we do this while x is greater than or equal to 1. So at x equals 0, we don't do this. And we decrease x by 1 each time through the loop. Now that we know that x is like 10, 9, 8, 7, 6, 4, 3, 2, 1, we have to turn that into the right argument. So one of the things about shell expansion is indirection. So if I have this and I do this, of course I get def. But what if I want to treat ABC as the name of another variable and then get the result, the value of that variable? That's what this exclamation point does, indirection. It's essentially pointers for shell. So here we look up ABC, which is def, and then because of the exclamation point, we use def as the name of a shell variable, which is 42, and that's what comes out. So that works with positional parameters too. So I can use x is now like 5 or 4 or 9 or whatever, exclamation point, $ brackets. Well, that's just something like $ bracket 5 or whatever. It's the same thing. It's just we're indirecting through x. So this now pulls out the x positional parameter that's left after doing the shift 2. So that's one of the input file names at this point in the correct order from right to left. Now we got to transform these to pull out the part number, increment the part number by 1, and then stick it all back together again. So another thing with shell, it's very important when developing shell scripts and so on that you kind of develop the pieces and test them out independently. So like, let's simulate this code. We're trying to make this code work. We're trying to program this thing or something. We're want to try to see if its parts are actually working right. So I'll set x equal to not x file. I'll set file equal to one of these file names. All right. So here's one of the file names. Okay. So file will be one of those things. So what is this like? What is this thing going to do? And part digits is going to be 2 in our case. So what is this crazy business here going to do? That's nice. Hey. Okay. Let me. No, okay, we're about to take a break, but let's. I messed up a little bit here in that I made FLE be one of the file names after transformation, but remember before transformation, they don't have the .fastq on them, So this is one of the file names like before transformation, which is the situation of the script. Okay, so then what does this business look like? Well, you see what this does is it's basically ripped off the part number. So how does that work? So, you know how this works. Well, then you can put :A, :B, where those are integers. And this now it says, take the substring starting at A and going for B letters. Okay, so the shell counts from 0. So that's the start at the start of the string. This is the length of the string, 23 characters. We subtract out the 2 characters that the part number is. And so we go from 0 to 21. So that is the substring without the 2-character tail. So that's basically the part to the left. Then you can imagine how this next one works. This has got to be the part number itself. That's the 2 digits at the end. So that's if you omit the ending position, it just goes right to the end. So we just have to tell what the starting position is. And the starting position is the last character minus 2. So we're doing good here. We kind of got the parsed out into the part before the part number. We got the part number. Now we need to add 1 to the part number. So unfortunately, we'd like to do something like, we could just do shell arithmetic, right? So here's the part number. We'd like to just do something like that. That looks like that would work. The problem is that the part numbers have a leading 0. And if the part number happens to be 7, you're going to see, oh wait, that works. Oh, yeah. 8 is going to be the first one in trouble. Yeah. Why doesn't that work? This is probably a missed feature, but in days long past, base 8 was much more important, octal. And because the shell is so old, the shell understands octal. And the way you tell whether a number is octal or not is whether it has a leading 0 or not. And so 0.7 is really an octal 7, but that's actually the same as a decimal 7. Maybe this would be the other surprise, 10 plus 010 plus 1 versus, well, 10 plus 1 is going to be 11, no surprise. But 010 plus 1 is going to be a surprise. 10 plus 1 is 9. Why? Because 010 is octal 10, which is 8. And 8 plus 1 is 9. So here's like some of these little gotchas that can happen. So the leading 0 is bad for shell arithmetic because it activates octal. So awk luckily does not do this kind of insanity. And so we will do the +1 in awk. So this business uses awk to add 1. I need to close and 10 there. And the reason we have to go to that much trouble is because we have leading 0s on the input and we'd like leading 0s on the output. And awk has the printf % like 5D. This will make the output be 5 digits 0 padded on the left. So we're using awk for 2 reasons: to avoid the octal activation in shell with leading 0s on the input, and we'd like to have leading 0s on the output, and awk makes both of those easy. So that's basically a fancy way of doing +1 that respects leading 0s. Then we can just move the file name to the part before the part number, the new part number, and then add the suffix that we want in the end. And that's how that stuff works. But now that we've looked at the command page to split, we don't really need to do this because we could have just added some more command line options to split and not had to do this. But anyway, it does show you how you can take strings apart in the shell, which is important to be able to do in general. Okay, so it's 3:30. That's another good place to stop. We've already showed the purpose of local with this. And so maybe we'll show just this to complete this logical thought, and then we'll stop. So now that we know about indirection, you can actually use indirection to do, I'll just show this because if you kind of get into programming in the shell, this is kind of useful, but how do you write a shell function that kind of modifies a variable in the outside? Well, if the shell function knows the variable's name already, you can just do it. We saw that, right? That was just like add a equals a plus 1, right? So that's this one, right? So a equals a plus 1, and this was affecting the outside a. But what if you want to sort of pass the variable in as an argument to the function? How does that work? So, like pass in a or b or whatever into the shell function. The shell function doesn't know which one. It wants the variable as an argument. How does it change sort of a variable that comes in as an argument? And the answer is to use the indirection feature, basically, to do that. So I'll paste this and run this code, and you'll see that it matches what's on the slide. So we can just look at the slide. So the idea is that we're going to set like a to 5 and then call bump a, and then a will be 6 on the outside. We'll set b to -3, bump b, and b will be -2 on the outside. So this bump thing is actually able to modify the outside variable, but the variable is coming into bump via an argument. So how does that work? So this is basically real pointers for shell. Pointer is the variable's name, a or b, say. And then, of course, pointer will be a or b, but that exclamation point will mean $! Pointer is the value of a or b. So what we really want to run to increase the value of a or b is to run a command like let a equals a plus 1 or let b equals b plus 1. So here's a double quoted string that actually expands to the right shell command to do what we want, right? So this would be like let a equals a plus 1 or let b equals b plus 1. There's a shell command called eval that will take its arguments and run its arguments as shellcode. And so that's exactly what we do. We compose the command that we want to run as a shell command, but then actually run that shell command. So here we're running dynamically generated shellcode. That's how we can basically emulate this sort of manipulation of variables passing into the pointer. So that we're not going to have any application for this in this workshop. I just thought I'd throw that in because it kind of shows you more how like you can kind of do normal programming, like real programming things, even in shell. Okay, so we'll take a 5-minute break there. Okay, so yeah, shell file name patterns. We could mention a little bit about those. So you probably know star. That's the most popular one. Star matches. So there's two kinds of patterns in the Unix world. There's shell patterns, which are called globs for silly reasons. And then there's regular expressions, regexes. So things like grep and so on take regexes. Default by shell default, shell does not speak regexes at all. Bash has regex extensions that you can turn on, but by default, doesn't understand regex at all. Shell does globs. So while some of the syntax looks the same, like star is both a regex thing and a shell pattern, shell glob thing, they mean completely different things in the two worlds. So, okay, so now we're talking about shell globs as patterns. What does star mean? Any sequence of characters whatsoever. Could be the empty string, could be a single A, could be a whole word, doesn't matter, anything. The other character that is part of shell glob is question mark, and that stands for any single character. The other thing that can be in a shell glob is a bracketed expression. So you can do like 0-9, and that means any single character between 0 and 9, which means between 0 and 9 in the ASCII order. One of these columns is that. There we go. Anyway, so 0, whatever. ASCII is sane in the simple region. So 0 to 9 means exactly what you think. A to Z means exactly what you think. But capital A to lowercase z would also include these things. Anyway, don't get too crazy with your dash ranges. That would be an alphanumeric character. That would be a comma or a hyphen. Anyway, so you can put those together in any way you like. So now some of these cases I could have simplified. I don't really have to be, let's see here. So what is that pattern going to match? That's going to match all the parts for both samples, okay? Because that's srx star, head, part, double question mark, two digits for the part number. That's going to match all of our parts. Now, because of the complete list of files that exist in this directory, I don't really need a pattern quite that specific. that would have been sufficient, right? I could probably even get rid of some more. I mean, you could even get rid of more like that, right? Or possibly you don't even need the head, right? You could probably just do that. And you don't even need the xrx even really, right? Like, that would have been enough. Like, one of the things you learn though with like the shell patterns and especially star is it's better to constrain those a little bit than to not, just so that you don't get surprised. Like, yeah. Oh, the star, yeah, you can do A star B star C star D, and that means, yeah, exactly what you think. Now, Bash even can do regular expressions, which are more powerful. The star, question mark, and bracket they don't always let you do what you want. I won't talk about it, but you can actually turn on, even for file name expansion in Bash, you can turn on full regular expressions and really control what. But most of the time, the star and the question mark and stuff, they're enough. But I do like to sort of vaguely specify the pattern, just so it also just sort of helps. You look at this code later, it helps you because without knowing what's the directory contents, you don't know what this expands into. So if you have some of the pieces as to what this really was intended to expand into, when you come back to this code and look at it later, it helps you know what the intent of this thing was, because, oh, I can really look at this and say, oh, this was all the parts. Whereas if I had just something like, star 0 to 9, which may actually be enough, now I look at this 3 months from now, I'm not going to know exactly what that was. So that's the logic behind kind of overspecifying the patterns a little bit. Is this good? Intense, but so take like the shell variable expansions, like $x. But you've already seen that we can do things like $%, X%, remove suffixes, or X:3:5 and start putting up pieces. There's like 30 operators you can put like there to like, you want to remove the suffix, you want to remove the longest suffix or the shortest suffix, you want to remove the prefix, you want to do a textual substitution, you want the substitution on the leftmost part, and alt, you want it substitute all the time here. They add up. I mean, so it's like so already just to like completely explain like what $ variable can do is like 20 slides. And that's just like one tiny aspect of stuff. So, but so this is kind of what I say, like you kind of learn that as you go. So like you're writing a shell script and it's like, that you need to transform a file name. And maybe you're not sure how to do it. Well, that's now like, oh, I need to change WT to wild type. How do you do that? Well, that's when you Google. You say, shell variables, string substitution, whatever, and find out how. Even at that moment, how to do that. It's not really a great approach to like, oh, I'm just going to sit down this week and learn all there to know about shell, and then I will proceed after the point learn everything. It doesn't. That's maybe you can make something with it, but it's not really. Yeah, so the very first slide, or one of the early slides, I think it's like why shell or why more shell. Here, more shell. Click this link. Especially go to the end guides and look at some of these. And these days with Google and ChatGPT and stuff, you can learn on the fly easier than ever before. There is a man page and a documentation for bash. Here's the man page. I'm going to hit space at a rate of about 3 a second, and you'll be able to see how long this man page is. Oh, it's less than 4,000 lines with a wide window. So you've seen it all. I could just hold on. Here you go. You want to learn everything you should know about shell? You ready? It's like Neo with it, learning how to do kung fu, right? Here you go. All right. All right. Now you know kung fu. Okay. Gotta find out where we were. Here. Okay. Blah, blah, blah. All right. Where were we? Oh, we want to gzip these things now. All right. But now we got 42 of them. Okay. So now here, like, how can we gzip them out? Well, I mean, the simplest command would be something like this, right? I'll put an echo in front just so it doesn't run it, but you see what it expands into, right? So what is that going to do? That's going to gzip. But gzip is a serial single core program. It's going to open up the first one, gzip it, and only then start the next file. It's on a completely serial process. This would be very slow and only use one core. Well, how about we use the tricks that we learned? We'll do a for loop over all of them and launch a gzip for each one of them and then background them all and do them all at once. Now, that is 42, and that's getting to the point where it's a little scary to kind of do that. I'll actually start it because, I mean, this is bad behavior on Hoffman because what have I done? In my QRSH, I kind of said I'm only going to use 3 cores. Well, not right now. I'm going to use 42. Whatever. What did we say? This machine has what? This machine has what? 48. Is there any? And is there anybody else running on this thing right now? Oh, there are people running on this thing. Yeah, there's some Pythons and R's, and I don't even know what Slim is. Julia. So it's a question of how much bad behavior do I want to do right now, right? So I'll start it, but I'll kill it pretty fast. So the kill will be a kill jobs-p, right? So that'll kill all my background stuff. But I'll start them for a moment. But this isn't good because I said I was going to use 3 cores, and I'm about to try to use like 42 of them. So bam. So I just started 42. They're all. Oh, stop. Anyway, let's get rid of them all. Oh, this isn't behaving like I'm expecting. This isn't going well. All right. I'm going to kill my session. I have to see if I've got any processes left over. No, I don't. The node's clean. Okay, good. All right, I'm not going to do that again. That did not work the way I thought. That should have just started them all running in the background or whatever. I don't know if that's interacting at all with the queue system having a resource constraint that I can't start, so many at once or something that on your local machine, that would have been fine. Your machine would be pretty well loaded at that point. But anyway, that's not a good way to do it because it's too uncontrolled. These are extremes. This is do them all one at a time. This is do them all at once. But when and what if this was not 42 parts, but 1,512 parts? There's a point where this doesn't work. So we need a way that's more controlled, something that can regulate the number of gzips running at the same time. So that if, for example, we have 3 cores to us, that start the first 3, but then each time one of them finishes, start the next one that we haven't done yet until they're all running and then they're all done, right? We need but that's like a complicated business. We need some program to manage that for us. But if we had such a program, then we could do this nicely. We could efficiently use our 3 cores. So there are 2 such programs, which are very common and very powerful. So the first one is called xargs. Let me get back to the directory that I want to do in. All right. So how, xargs has a man page or whatever, but you're going to find it, hard to kind of, learn xargs from that. So xargs works basically like this. You have some process that produces file names, doesn't matter how. And then you pipe them as standard input into xargs. And then xargs has some options, which we can talk about. But then at the end of the xargs command is a shell pipeline. I mean, it's basically what you're going to do, it'll run with, like one of the options that it controls how many of the inputs go into each pipeline that it runs. So we want each of the file names to be launched a separate command. So -n1 means every time you get one file name on the input, you're going to launch one command on this side. So that's a simpler situation than like n2 or n3 or something, just like the n=1 situation. So I mean, for us, that's going to be a gzip. Something like this. Okay, so I mean, these options you don't even really need. This is verbose, so the gzip tells us when it starts a file and the % savings when it finishes a file, and the best means work very hard to compress the file at the expense of using more CPU time, or fast would say don't use as much CPU time. You're going to get a slightly bigger file out, but use less CPU time. The double dash isn't really necessary because none of our file names will have hyphens at the front or anything like that, but it's just habit me at this point with xargs and stuff. Because in general, xargs, you're doing stuff like some find all these files in the file system. It could be many files. You generally want the command on the right side of the xargs to be as safe as possible. So anyway, this is just sort of like a safe version of gzip, but for our purposes for this example, that would have been fine too. So anyway, what xargs is going to do is, it's going to read a file name and then start this, and read a file name and start this, and so on. And the default for xargs is to just do one at a time. But xarg can also parallelize. You put P3 here, say, and that says run 3 at a time. It does exactly what we were talking about in the red text here. It'll start. I mean, if you put in less than 3 file names, they'll all just run right away. But in general, if you put in lots of file names, it'll start 3. And then as soon as the first one of those 3 finishes, it'll start running the 4th. And then the next one that finishes, it'll run the 5th, and so on until it reaches the end of the input list of file names. So all the rest is just details about how you construct the file names and so on. And do you worry about file names that have spaces in them and so on. So that's what all this business is here. So one way to generate the list of file names is to use ls. So we're used to using ls to see long lists of files like this and so on. But one of the options that ls takes is ls-1, which just makes each file name on its own line. Okay, so that's kind of convenient. So that's basically this. We could have also had the shell just do like an echo and then like transform space to new line or something. That would have been another way to do it. Anyway, so those would both work. So we could do something like this and then pipe that to xargs, n1, p3, gzip. That would be one way to do it. It's to deal with white space. I mean, we don't have to. None of our file names have white space, but a good habit with xargs is to at least deal with white space problems. So in that case, the way it works is that what if you have a file name with a space in it? If you have a file name with a new line in it, that's a different story. That's even worse. I'm not going to tell you how to deal with those right now, but obviously, if a file name has a new line in its name, that's going to cause confusion as to where one file name starts and another ends. But new lines in file names are really rare. That's almost always a mistake. But spaces and file names are not that rare. So those are worth defending against. So how would you do that? Suppose I've got I want to gzip file names like this. Whatever. So we got some file names with spaces in it. ls will have no problem constructing a list of those guys, so on. But how would we get those into xargs? Because xargs is very sensitive to all white space. So the answer is instead of using new lines instead of using white space to separate file names, you can tell xargs to use null characters to separate file names. So this transliterates new lines to nulls, which isn't going to look very good on the terminal. You can't even see what happened there. But if we kind of expand that into binary, you'll see that here is the first file name, ABC space DEF null 00. GEF space GHI null. Whereas without the TR in there, it's ABC space DEF charge return, new line, well, new line, DEF space GHI, new line. Anyway, so the null is a much more useful separator character because it is in fact the one character that cannot in any Unix or Linux anywhere ever possibly be part of the file name, because the C programming language uses that as the separator to mark where strings ends, and Linux and so on is written in C and so on. The whole thing is C at the bottom anyway. So file names, the one character that can't be in a file name is null. So if we use null to separate the file names, that's guaranteed to handle everything. Here we're using ls to generate the file names, and ls uses new lines to separate this thing. So we'll be confused with file names that have new lines in them, but those basically don't exist. So this is almost full generality. So anyway, so we transliterate the new lines to nulls, and then we tell xargs -0. The 0 means your file names are separated by nulls. This is a good kind of pattern to have because it basically works for every, almost every kind of file, whatever. It defends against every kind of white space except new line in a file name, which is so rare it's not even worth really talking about. So, this is basically kind of a safer form, something like this, and that would be pretty safe. And this you could use even with a fairly nasty list of files, and it would gzip them all, no problem. Oftentimes, you need to do find the files in more than just the current directory, like a whole tree of files. And then the find command is really helpful. You may have seen find in workshop 1. Here's the man page for find. You'll probably find that very hard to read. Tutorials on find on the web and so on are probably much easier to read. But with find, you can do things like say what directories you want to look in. Like I could say, start looking in from the current directory, find me all the files that have an A in the file name or something. Well, that's, here we go, like that. And it'll be a pretty long list there. Or ABC. There's a few. There's my ABC space DEF and so on. But here you see the find search down through this whole place, which has some very long nested directories of Python stuff. Anyway, the point is that whatever it is, ls find, spitting out file names, find can do a print 0, and that makes sure that find file names come out with a 0 separator directly, and then you can pipe in x0. So this is a very popular kind of pattern. So you'll see find, some places, some specification of what to filter in those places, and then a print 0, xarg 0, and then some command over here like gzip or whatever, and basically find all the files that meet these specifications and gzip them, and gzip them in parallel. Okay, so this is really kind of a powerful set of things to combine, because now you're not limited by number of files. That could be 50,000 files, no problem. I mean, if it takes a while to gzip them and you're doing 3 at a time, it'll take a while, but it will go through them and finish them. Whereas the 4x in something do gzip, x background, whatever, that's going to be good to maybe a couple dozen at most or something, right? But you get into the hundreds, that's no good. Yeah, these are very powerful, these kind of patterns. ls to xargs, find to xargs, very good. We could run any of these and they should work. I'll run this first flavor here, and we'll put that in the background. And you see, I've got the -VV verbose for gzip turned on. So you'll see here that gzip has started xargs has started 3 of them, right? So you see, where are they? Where are we here? So that is the first one started, the second one started, and the third one started. Everything is kind of mixed up here because the gzips are printing out their progress information, but they're all interleaved together. So that's why we kind of see things, and they're going to all print out the progress as it kind of happens while we're kind of doing things here. So we've got this command pipeline running. The PS will show us this. So you'll see here that we've got the ls is done because the ls was so fast and generated the list of file names, it's not around anymore. And then the tr took the output of ls, and that's not around anymore. So the only thing left is xargs, which is managing the launching of the gzips, and 3 gzips that are running at a time. And now you see that some of the gzips are finishing, and then it's immediately starting another gzip again. I actually have the verbose on to xargs. So you're also seeing this, which is the command that xargs is running. So there's a command that xargs started, and then this gzip command, this one here, this is its first line of output, which is the file name that it started working on. Then this actually is a finish message from some other gzip. I mean, here it looks like it's all one line, but it's really this was printed by this guy, and then there was a pause, and then some other gzip finished, and then that's its finish message. So I mean, things are just all interleaved here because we got three running in parallel, but you kind of see the flow here. If I check the PS again, you should see that we still got the xargs chugging away, but the list of gzips now is different, right? So actually now 1, 2, and 3 finished, and now it's 4, 5, and 6 that are chugging along. And actually, because all these parts are about the same size, and they're all from reads that have the same statistical distribution, they're all, and we synchronize the start of all three of them at the same thing, they're all kind of running about in sync right now, right? As we get towards the end of this list, they should become a little more dephased, right? Because there'll be slight variations that'll add up once we kind of go through a few rounds, but they're still sort of mostly, you see them mostly finishing in little batches very close to each other in time. And so I'm starting, it's just because things are so uniform here. But in general, these things will be starting and stopping just kind of completely randomly. So we're up to 7, 8, 9 on the first sample as what's in place. Yes, I think there's Q alter. And that modifies the resource requirements of a job already in flight. So we have to talk about the queue system. We haven't done that because we haven't done any queue jobs, but actually QRSH is already a job. So if I do qstat-u focus and show me all the jobs that I need, you'll see that I have one job, which is running, which is QR login is the name of it. This is my interactive session. You see that it's on node 7041, which is the node I'm on right now. And I have 3 slots, that's 3 cores. 3 cores on 7041. And this was started at May 2 at 1550. That's 250. This, remember, it's not like 130 or 140 because I remember I had that explosion with the tried to do all the gzips, and so I killed the session, started a new one. So this was started at 350. Yeah, 350, like 2 minutes ago. So yeah, so this is the job that is currently scheduled to, well, this job is 3.5 hours from the start point because we asked for 3.5 hours. So this is going to end at 1850. Can you change the ending time? Yes, that's Q alter. I'm not going to tell you the syntax for that, but it would involve things like the job number, which is here. And then you have to change the resource specs. And now you have to kind of say the new time. And then it has to get filtered by whatever the Hoffman administrators allow you to do. So I'm not sure if they allow a time extension, or maybe they allow it up to a day or whatever. But the system has facility for this, but whether it's all allowed in a particular situation or not, it's kind of complicated. But yeah, you can sometimes extend the session. But that's why in general, once you get to large numbers of jobs, you need indicators that things went well or not, because you're not, you don't always know if it's going to make it to the end or not, right? I mean, you may launch a bunch of stuff and then you have to go away or disconnect, and you start stuff at school and then you go home and you reconnect to the internet or whatever. I mean, things can happen when you're not looking. If you're not watching every line that goes by of 1,000 jobs. So you need indicators. One of the big things we'll talk about once we start all these parts aligning is how are we going to know whether they all worked or not. Okay, where are we now in terms of progress? So we're on 16, 17, 8. This is going a little slower than I think it ran yesterday. I made it, I kind of finalized the slides yesterday. So anyway, we can talk about GNU Parallel while we're at it. So the other major helper program that does a similar kind of thing as xargs. I mean, it does everything xargs does and like a billion things more, is GNU Parallel, which is the web page for that is here. And there's all kinds of tutorials and stuff on this thing. There's like a tutorial and stuff or whatever. But in its most basic form, it basically is xargs with like exactly the same arguments. Even it understands xargs command line options. Now it is on Hoffman, but it's not part of the default path of Hoffman. So if I ask for a parallel, it doesn't work because it's not there. But you may know about the Hoffman module system, which has a whole bunch of pre-installed software. It's just not all active at the same time. One of the reasons is because they have multiple versions of stuff installed, and you need to say which version you want, and so on. So there's the full list of the current stuff, and somewhere in here, these are sorted by name. So somewhere in here is Parallel. It's in the module system. And so you say module of load parallel. And now we can use parallel. Yeah, so we have these without all the, the nice thing about the verbose messages is that once in a while you see some progress here. The nice bad thing with progress messages is that they're all just kind of injected into your terminal session randomly. So it can be kind of jarring. I mean, sometimes the watching with the PS, and seeing what's kind of alive right now is better than the progress, these progress messages. And get rid of the progress messages and just start the xargs and stuff without all those verbose flags. Actually, we're on the second sample now. We're on 1208. We're halfway done basically. So now that we have parallel loaded from the module system, it's around. It has help page and so on, and a man page, I think. And it has a lot more features than xargs, but in its most basic usage, it's just xargs with the same option. So we can just basically take one of these command lines with xargs and basically just replace xargs with parallel, and it'll work. And it actually, one of its nice features, you can add like an estimated time of arrival bar. So, maybe I'll stop while we still got a few parts not gzip yet. So I'm going to kill that guy. And you'll see here that some of the parts are gzip and some aren't. And the way gzip works when it's interrupted is that if gzip is interrupted in the middle, it actually takes care to delete the partially gzipped output. So and the gzip doesn't delete the input file until it's completed the output file. So you see here that we kind of are in a clean state. It was probably doing 4, 5, and 6 in flight when I killed it. But you see, we still have 4, 5, and 6 uncompressed, and we don't have a partial 4, 5, 6 compressed. So gzip cleans up with itself if it gets interrupted. So that's very nice for right now, because it means that I was just able to kill it kind of anywhere, and we still have a consistent state here, where some of them are gzipped and some are not, but we don't have like some missing, and we don't have like partial gzips, right? It's clean. So I could start one of these again, and it would work on the parts that haven't been gzipped yet, because my file name pattern does not match .gz guys, right? So we're okay to restart the ones that are left using whatever pipeline we want. So let's try using parallel now. And here I'm not going to put this in the background, because with this ETA progress bar stuff, it's going to make a text progress bar, and I need that in the foreground to be able to see the progress bar. So there we go. And you see parallel is a lot more verbose. And it's really insistent that if you ever use this thing, you better cite me in your papers, which is fine. But you see here at the bottom, the estimated time of arrival and stuff. Now, it has no way of knowing the progress internal to each command. I mean, all it can see is I started a gzip and when a gzip finishes. So it doesn't have visibility inside the gzip about what percentage of the way each individual gzip is through. All it can do for timing is measure the time the individual gzips are starting and finishing. So until a couple of gzips have finished, it has no way to even estimate an ETA right now. But eventually we're going to get, we got 3, this says it's got 3, we got 3 running on the local computer right now, but none of them have finished yet. 18 pending. But until some of those finished, we can't estimate a time of arrival, because the only thing we can do is see when a gzip starts and when a gzip ends here. So we'll have to wait until somebody's finished to see a bar. There we go. I still have all this gzip VB in here, so it's still making a mess, but it's okay. It gives you actually a little history of what those lines were. So anyway, so now we've had some finish. So you can see here that it said, what is it? So it'll stay, it still doesn't have much data. It's only seen like 3 at the finish. It'll stabilize a little more once it has some more done. But there are now 15 left and so on. Anyway, you get the idea. You can play with this stuff. But that's pretty cool. So these basically are almost kind of like QSub on your local machine or on a single interactive session without all the craziness of QSub. Okay, so QSub is not Sun Grid Engine, Univa Grid Engine. It's not something you're going to install on one of your personal machines. You could, but it's really complicated, and so on. But in terms of how you use it, typically, xargs and parallel basically give you almost all the functionality QSub that you would use, and you can use it easily on your local machine. I mean, xargs is going to be part of a default install almost everywhere. Like here's on my Mac laptop, it's part of the base OS X install. it's going to be part of every Linux base install and so on. GNU Parallel might or might not be part of base install, but on Linux, it's going to be trivial to get it. It's in your package manager if it's not on there by default and so on. Anyway, and they basically let you have a large number of jobs that are run in pieces that are matched to the number of cores on your machine. Right? So it's basically like a little cluster, a little one node cluster for your machine. So that's great. The only so the next step beyond this is to start using the Hoffman clusters, the queue system, right? So in terms of the evolution here of how this we're gonna, what we're gonna talk about, and we're getting close to 4:30 here. So now we're talking about stuff we're gonna do in day 2. So the next step is to start our BS alignments, and we're gonna start doing those by individual jobs with QSub, because that we have to make sure we know how to use QSub at all and so on. And so the simplest way to QSub is to just use one QSub job per part, but that doesn't scale too well. That'll be okay for 42 parts, but that doesn't work too well for 1,000 parts. And so once we learn QSub for individual parts, then we will learn QSub for big numbers of parts. So that's what we're building up to. So you kind of seen kind of the levels of parallelization are sort of percent sign, right? So shell percent, we saw that a bunch of times today. And that's good for small numbers of things, but very, very convenient to use because it's like literally like change semicolons to percent signs or add a percent sign at the end of something. And it basically, it's like magic, it works, but it doesn't scale very well. But it handles small problems very conveniently. Then the next level is kind of like this like xargs parallel stuff. And that works for even large problems, but you're restricted to one machine. So it works okay for your local machine or one interactive session, one node, but it's not going to let you use multiple machines in the cluster that well. So then the next level will be QSub jobs, which are good because they'll let you use multiple nodes on the cluster and they keep track of. It'll kind of do the xargs parallel thing, but instead of restricted to one machine, it uses the cluster. But that still has problems when you get to thousands or hundreds of thousands. And then the next level of that is how do we kind of deal with what should be, what you'd like to be is thousands of QSubs, but you can't do thousands of QSubs. And that's job arrays. So that's kind of the escalation of things. You kind of just choose the level for this direction that gets your problem size solved. So, but in general, for like these large line of problems and so on, you're going to do how to do the job. So that's what we're aiming to reach at day 2, is the job arrays. Okay, so we've got we got 15 minutes left. So if we can get to, okay, we've almost got all the parts. Are we down to like 3 left? We got like 3 left. All right, we're almost done here. So the first thing we'll talk about is just how to, now as I said, we don't care about the biology here. So like the choice of aligner and the parameters of the aligner and cleaning up the reads first and make, whatever that, we don't care about any of that. We just want to run the aligner as just a placeholder tool for serious computation. So the first thing to do is just to make sure we've got an aligner that we can run on a part, even just one part at a time, because we can't do any of the fancy stuff until we can make it run at all. So I think in the 15 minutes we have, we can probably just get to the point where we can just run the alignment on one part in an interactive session. And then so day 2, we can start with that at the base. And day 2 will be about then being able to do that on all 42 parts and scaling it up appropriately. But we need to be able to run, even in an interactive session, the alignment of a single part before we can even do any of that stuff. So let's get there. Okay, good. Okay, so I think we've got all the parts gzip now. Let me clean up some of these stupid files. There we go. Okay, so we got our parts ready. They're gzip. This would be the end of the split stage. Okay, so now we have the data in pieces. Now we need to start working on the alignment of the pieces. But before we can align all the pieces, we need to learn how to align one piece. So how can we do that? As I say, we're not too big into biology here, but so we're just gonna, we don't have to worry about. This is just sort of like this now would come from. This is now stuff that comes from workshop 6, basically, which is our bisulfite workshop. And so we're going to use bsbolt. That's a Python program. So we need to activate Python in our interactive session. We need to install bsbolt. We're eventually going to need SAM tools because SAM tools will be the thing that lets us merge BAM files and sort them and so on. So anyway, so here's the commands that are going to do all that. Oh, before I do that, what was the gain of the gzip? If you look at the file sizes before and after, the slide has, back a few slides, has 6.1 gig is the before size. It's now 1.7 gig. And so that's a big space run. We just divided the space by 3.6. And so this is why you really want to keep things gzip in big projects, because that's a big win, right? You know, we're talking like more than a threefold savings in disk space to gzip those intermediate parts. And yeah, when you're pushing terabytes, you know, suppose you've got, I think we cut the data down I think by about 40-fold when we did that 10.1 million read subset. I think that was a divide by 40. And then maybe instead of 2 samples, you've got 20 samples. So that'd be another factor of 10. So in a real project, you might be 400 times what we're doing as a live demo. And so now you can see, you can hit you know, that gets you close to terabytes then of disk space once you start having a few copies of that around. You know, you've got the reads, and you've got the split reads, you've got the alignments, and you know, the part bands, and you've got the single sample band. You can see where disk space can kind of really become an issue at any moment, because especially because in Hoffman you've got quotas and stuff, right? So you don't have infinite space. Okay, so anyway, so how do we align a single part? Well, here we're just pulling from workshop 6. We're gonna use Python version 3.7.3 here. That's in the module system in Hoffman, so we're gonna activate the Python. And then we're going to use something called virtual environments in Python, because I don't know if you've already got Python stuff going on in your Hoffman. And some people have real work that they're doing and they have a Python state set up and you don't want to disturb that. Or some people have played with Python a lot in the past without knowing what they're doing, and they've got Python in some weird inconsistent state with package manager having some pieces at some versions and some in others, and nothing works at all. So anyway, so the point is, I don't know what state your Python is in Hoffman right now. I don't want to change it because you might be using it for real stuff. It might be broken, so it might not work for my stuff. So the answer is we need to isolate. Let's just leave whatever you got going on over here, and we're going to do our Python stuff over here, and they're not going to have anything to do with each other. And the way you do that is what's called a virtual environment, which is a Python 3 module these days called vn. And what you can do is you run a command like this. You say Python 3-m vn, and then some directory. And what this does is creates a new virtual environment. You just run this once each time you need to create an environment. So like day 2 tomorrow, when we go to use this, we will not rerun this command because we're going to use the environment we already exist in. At the moment, we're creating a new one. So this is vn creation. And what this does is make a whole bunch of files and stuff in that directory, but it's basically kind of looks like a clean Python installation that has nothing installed. And so that's what we want. So that'll take a little bit because it's making like hundreds of files and a whole directory tree and whatever. And we don't really care what's inside there, just the effect that it has. There we go. We can peek. All it's done is it's made this directory vn, and there's a whole ton of stuff in there, including a bin directory where you see stuff like Python and Python 3 and so on and whatever. So now to use that environment, you have to have the module. So that is a Python 3.7.3 virtual environment. So before you use that virtual environment, you have to have the module load Python 3.7.3 already active. It's a VM on top of Python 3.7.3. So you need this environment active first. But then to activate the virtual environment, there's this activate shell script inside the bin directory, inside the VM directory. You need to source this shell script. You can't run it as an ordinary shell script because this shell script wants to play with your path and other environment variables in your interactive sessions shell script. And if you run this as an ordinary shell script, the way ordinary shell scripts work is that a new shell is created for the shell script runs in the new shell, and then the new shell exits, and now you're back in your calling shell. Well, that new shell would have its path changed and stuff like that, but as soon as the shell script died, all those changes go away because this thing cannot change the environment back here. So what we need is that script to actually modify our stuff in this shell. That's why we need to source that script, which essentially just means take all the commands in that file and run them in my current shell as if I typed them into the interactive session directly. And that way it's able to affect the path and all this stuff in my current shell and so on. So that's why we source this thing. So what's this directory spelling? Bin activate. So that's why we source bin activate, so that it's actually not making a subshell to run the commands. It's actually running the commands in my present shell. Okay, and now you'll see that actually my shell prompt changed, because one of the things it does is change the shell PS1 variable, which is the prompt string. So you'll see that this PS1 variable actually has this now as a prefix, and this cocus-u is the cocus --u is the user, -h is the host, -w is the current tail part of the current working directory. You can actually read the man page and make your own prompts with all kinds of data packed into them or whatever. That's the PS1 variable is where all that prompt stuff happens. One of the things that this script did is change PS1, which is why our prompt changed. That's not really anything besides informative. What it's really done is do things like path. So path now actually has at the front of it the bin directory of that VM in the front. So that now when I do like Python 3 and stuff, it's actually going to pull from that virtual environment stuff. And this is why now when I look at what modules are installed, it's nothing, because this is a clean environment. If I look at my, let me do this in a new Hoffman session. If I did that Python module load, Python 373, and then if I did Python list, you'll see that in my normal Python environment, I got a zillion things loaded. So this is an example of something I want to isolate from for the workshop. So we're in a clean situation here, and now I can install a BS Bolt and have pretty high confidence it's going to work, because it's not going to interact with some broken package state or version conflicts or something that are whatever you got going on in your main Python. So this is now going to install BS Bolt inside the virtual environment, and I don't have to really specify anything because the VM has made sure that Python 3 is now the path and pull it from the VM, and all the defaults inside that Python 3 are now pointing into the VM and stuff. I don't have to tell the pip or install anything that I need to install this into my personal directory somewhere, whatever. The VM takes care of all of that. Everything's redirected inside the VM. So it's great. So here we're installing BS Bolt, and if BS Bolt has a bunch of dependencies and those have got to be pulled in and so on or whatever. But anyway, this is great. In fact, pretty much for any serious project that uses, say, Python or something like this, I really recommend making sort of a VM for that project that isolates all the Python stuff for that project in some pile. Because eventually you'll end up using so many programs and so on, they'll have mutual incompatibilities where you literally cannot have all the programs you want to use simultaneously installed because they have conflicting dependencies on packages and stuff, or whatever. And the only way to do that is to make virtual environments and isolate them against each other. And so it makes sense to just sort of for each major project you do, make a VM that collects all the packages you need to work with in that project isolated. Anyway, so okay, we've got it finished. And so now we've got a BS Bolt available, and that's got a bunch of things it can do. And one of them is the align stage. It's got all kinds of options which we don't care about because, again, we don't really care about the actual insides of this. We just need to be able to run this thing in some form. So we'll take defaults for pretty much everything. We won't need SAM tools today. We're almost, I got 5 minutes left, but SAM tools is in the module system as well. And SAM tools has help and can do many things. One of the things it can do is merge BAM files. We'll be using that functionality to piece together our parts back into single sample guides later. Maybe actually we will do a day 3. We'll see how far we get tomorrow as day 2, whether we need a day 3 or not. But anyway, so okay, so we've got BS Bolt kind of ready. That's great. How would we actually align a part? Well, since we don't really care about all the command line options that change the way the alignment parameters work and stuff like that, it's really pretty easy. I've already given in this directory a pre-made index of the human GRC H38 reference genome ensemble release 103. In this directory is a pre-digested BS Bolt index for that human reference genome. So this is already kind of ready to go. This is something you have to BS Bolt index from the reference genome FASTA first. This is a fast index structure. It's really, BS Bolt really uses BWA, Burroughs-Wheeler alignment inside. BWA is an alignment program you're probably familiar with. It's a pretty popular one. This is basically a BWA index digested up, which allows fast alignment of the reads to the entire reference genome. It's just a computer science data structure that just makes the alignment go much faster. So anyway, but that's ready to go. The index is needed. And then, so then what does the actual commands look like? Well, so like, it's basically BS Bolt align. Then you have to tell it where the preprocessed, the indexed reference genome is with -db. And then we're using single-ended reads here. So then -f1 is the GZ FASTQ input file for main end one of the reads. And then -o is the base file name for the output band. It'll put .bam on the end of this, and that's where the output alignments go. And then -t is how many cores you want BSBolt to work with inside. Like if we were going to run it in our interactive session right now, we'd do -t3 because our interactive session is set for 3 cores right now from this. That's our scheduler reservation. Okay, so the rest of it is just sort of making shell variables to parameterize it so that it can work on any part that we have in progress. So here's the first part. This is the samples. Remember, it's like 7. They're stand in a 7-digit or an 8-digit. So this is like sample 1, part 1. This is the first one of our 42 parts that we need to do. So if we had a shell variable that was sort of like the name of the current part that needed doing in GZFQ, in GZFQ, and then we're going to need an output file name built from that. Right? Our usual pattern is sort of like some kind of, usually we need to build the output file name from the input file name. So we'll do that like this. So we'll take the ngzfq, we'll delete the .fastq.gz from the end of it, and then suffix on the end, BSBolt, DRC830, and 103. Generally a good idea in bioinformatics if you sort of encode to the extent reasonably possible all the information about how a file was put together into the file name, because that way when you do analyses like 20 times and then it's 3 years later and you're writing the paper and you're trying to figure out what parameters were that you actually used, the file names will tell you all the relevant information. So here we're saying we use BSBolt aligning to the GRC830 and 103 reference genome. I mean, there's more parameters than this. Like if you start changing BSBolt alignment parameters, you should probably embed those in the file name too, but whatever. That's just sort of a side thing, but so here's one of the file names that might come out. Like I said, BSBolt's going to put the .bam on the end. So one of the file names that comes out is going to look something like this. Okay, so that looks good. Here we're going to need to parameterize based on how many cores, because that's going to be set some earlier up in the shell script or something that eventually runs these things or something. So in the current session, that's 3. And that makes the command to run an alignment something like this. Okay, I could hit enter right now and that would work to align this one part in this interactive session. That would be okay. However, one of the big problems on Hoffman is choosing those resource requirements, right? How many cores? Well, that at least won't usually cause things to quit, right? I mean, if you ask for less cores and your program might run slower or something, more cores runs faster, okay, no big deal. But the other resource requirements are like time. Okay, well, that's, I mean, again, time, if it's too short, the job will quit. At least the job system will let you know then that it killed one because it ran over time or something. And time, if you give an overestimate and it's not too crazy, nothing bad will happen, right? But then the last one that you have to really give it is the memory guys, right? So, and those are the really nasty guys to figure out, because how much memory is BSBolt really using? It's like, no idea, right? I mean, and the problem with those is if you get it wrong and you give it way too small, the job's really going to die, like, in somewhere in the middle, and not going to make any progress. If you make it way too big, well, that at least the job would run, but now you're into the problem of reducing the size of the pool of available nodes that can satisfy the request. And now, if you ask for like 10 times the amount of memory that you really use, you're really going to hurt yourself in terms of like how many of those you can get going in parallel and stuff, because scheduler's going to have problems placing you and so on. And then if you're in the twilight zone, be kind of like the boundary between where things are like definitely going to fail and like way too much. There's this twilight zone in the middle where kind of like it becomes stochastic whether things work or not. And it starts depending on like the state of the node and how overloaded the node is, and like part to part variation and how much memory that BWA might need and stuff. So nasty stuff happens in the twilight zone in the middle. And at the moment, we sort of have no idea. Like at the moment, maybe we've never run BSBolt before. We have no idea. I mean, who knows? So one thing you can do is to like run one of them in an interactive session and then prefix something like userbin time-v, which is a resource monitoring tool, okay? What that'll do is it'll run this command, but then use the Linux operating system support to monitor resource consumption of this thing, and then give us a little report when it finishes. I should have run this like right away when we started talking about it, because it'll probably be done right now. Maybe I think I have the output on the slide, but yeah, okay, I have the output on the slide. So maybe, we're about out of time now, so we need to stop. But basically what time is going to, first, So we're running the first part for real in our interactive session right now, but we kind of are doing it with some resource monitoring that's going to give us a little hint about how much memory and stuff this thing uses. So we're going to see some progress information now, which is actually progress information from BSBolt and the BWA aligner that BSBolt runs inside. That's going to scroll by. That's this kind of stuff here in light here. But then BSBolt's going to give us some statistics information itself, like the stuff about the alignments. And then the end of that is going to be stuff from the bin time. And that's going to be stats about the resource consumption that this thing has. So the output is eventually going to look something like this. And we want to just check a couple things here. So one, BSBolt says like, oh, I saw 500,000 input reads. Well, that's a good sign because that's the right number of reads in the input file. 94% of them were aligned to the reference genome. Well, that sounds like good. That's a biological thing, but that's like, if that was like 0% or 10% or something, we'd be worried or something, or we'd have something to look into. But that looks fine. What about the resource consumption? So it took 4 minutes, 30 seconds. This is, when I ran it like yesterday or something. So, if I talk for another 2 minutes or something, we have a chance this thing might finish. But yesterday it took like 4.5 minutes. Maximum residence size kbytes. This is your indication. This is not, there's different ways to measure memory, and it doesn't always line up with the way QSub deals with memory and so on. Memory and memory enforcement and so on is a really complicated issue, but this is at least some hint about kind of what the order of magnitude might be. So this is in kbytes, 1024 byte units. So if we multiply that by 1024, that's bytes. With K and G, that's like 13 point something G. Is kind of like the maximum peak memory that this thing was using. Maybe you're surprised that it's that high, but it's not really that much of a surprise because it's got to load the reference genome index mostly, right? So if I look and see how big that reference genome index is, where's my directory? So here is the directory with the reference genome index. How big is that? 18 gig. So most of that index has to get loaded into memory. I think actually the FASTQ part of it, the actual sequence itself doesn't need to be read. So where is it? This BSB ref FA, that might actually not be needed. That's like 3 gig. So if you take the total size, where did it go? 18 gig minus 3 gig would be 15. Maybe, it's more like closer to 15 that it's got to load or something. And we're seeing like 13 here. Anyway, this is sort of where that 13 is mostly coming from. That's like reference genome index. Doesn't need a lot of memory to align individual reads because like the number of candidate positions for each read isn't going to be that big. And while this memory would scale a little bit with number of cores, it's not going to scale that much. Basically, BSBolt's memory usage is mostly dominated by the reference genome index. So it's good. So anyway, so this is not that unexpected that it would be like 13, 14, 15, 16 gig or something like that. But that gives us at least some idea of where to aim for the resource requirements. So that's quite helpful because 5 minutes ago we had no idea. Right? So now we could also have problems due to the fact that maybe our interactive session is too small. And in fact, actually, we are kind of skirting danger right now because our interactive session, remember, was 3 times 4, 12. We're actually a little over right now already, right? So this is already 13 and change, which is well over 12. So actually, clearly, Hoffman goes through moods and phases depending on what the administrators are into on the day. Hoffman is not really strictly enforcing the memory requirements right now. It's more of like a scheduler advisory in terms of like how much to kind of like block out and stuff. But it's not like instantaneously when a process reaches that memory size, it's going to die. The reason is because when they have that on, so many people complain, it causes so much problems that they went back to letting things kind of slide a little bit. But that actually causes other problems because it means that if other people's processes are overusing their reservation, the machine can be kind of overloaded. And then eventually the Linux operating system will run out of memory and then it may start just blowing up jobs on its own. And that may be blowing up jobs in a way that's kind of hard to detect, which actually we'll see kind of happen tomorrow. I want to show you, once you start running thousands of jobs and stuff, you do get kind of situations where individual jobs fail and so on. You need to be able to detect failure and then be able to rerun the ones that fail because you're not going to run a 13,000 part job cleanly in one shot. It never happens. Some of the nodes have broken hardware and stuff or flaky. You always do a big job. It takes a couple rounds. you need to find out which parts are still not working, and then rerun it and so on until you eventually get them all done. There you see the interactive session finished. So 500,000 reads, good mappability, and here's some resource info, 5 minutes. Here's our memory again, about 15 gig change, something like that. So, all right, so we're in an okay place to start for day 2. I mean, it's not where I thought it was going to end, but that's perfectly fine. We really have 3. I really aim for 2 days, but we have 3 days on the schedule, so maybe we will use the 3 days. So anyway, so day 2, we're going to start with how do we run. So now we've run it once in an interactive session. This would be very painful to do all 42 parts this way. So next thing to learn is how do we Q-sub these so that we can run each part in one Q-sub job. Then how do we do the real big stuff with job arrays? So that's what day 2 will be about. Okay, so that's it for today. I'll take any questions and then we'll see you tomorrow. Stop your recording.